my dearest little children. Hello, 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 hello. Just a second, okay? Am I audible? Yes, I'm perfectly audible. Hello to my cutest little champions out there. Hello Akash. Akash is correct time today. Oh my god. I'm really glad to see you over here. We'll just wait for some more time so that anyone who wants to join can come and join. By the way, today's target is a little bit refined over here. So today we have 3 chapters to do, right? And the first part we could only do sexual reproduction, flowering plants. Here we are planning to do human reproduction, reproductive health, and also one more thing, that is principles of inheritance and variation. Okay. Hello Pawan, I definitely remember you. Yes, I remember you attended the A series live. Okay, so this is how I planned, and basically as of now I'm directly going to go with the conceptual questions. What I have planned is we have the question bank of the previous year, so whenever we are sure about all the concepts, we are going to go ahead and solve this MCQs directly from the question bank itself. Okay, Manoj is also here. Manoj, how was your exam, da? You said you had your exam today. Today. Good evening. Okay, so I believe we are ready. So first of all, tell me, which is the first chapter that you want me to do? Is it human reproduction, or basically is it principles of inheritance? Reproductive health, one shot I have already made. I have just recorded and I'm back. So you can tell me like which one you want. If you want, we can skip reproductive health, because as I said, one shot is there, in that you find the answers. So Manoj, yesterday was my exam. I'll get 30 plus. Wow, wow, wow. Yes, ma'am. Please complete human reproduction first. Okay, let's go then with human reproduction. And the first of all, the things that I'm going to say is, here you're going to get 3 and 5 marker questions majorly. This is a general trend that was there. Now, in the case of your FA-1 examination, this trend can change, but the concepts which are important here remains the same. And the first very important thing that we have is, draw a labeled diagrammatic view of the female reproductive system. Okay, this is very important question, and I'm just going to go ahead with it. You make sure you watch me draw and make exactly like this. Okay, so over here for the human reproduction system, first of all, make something like this, a dome shaped thing. Okay, and over here you have something like this, a finger like projection and stuff like this coming right over here. Similarly, you would be having a same sort of structure up there as well, where you would be having finger-like projections, and yup, cool. Now further, we have to go ahead. Here also we would have to go ahead and make a structure like this. This is going to be the cervix. This region is going to be the cervical region. Over here you have the next part, which is going to be the vagina. My dear children, please listen to me. These diagrams might seem silly for you, but this is very important. Here is the vagina. What do you have over here? This is going to be the cervix that is going to be there. Now over here there is going to be a ligament, and attached to this ligament you have the ovary. Similarly, over here you have a ligament. Attached to this ligament you have the ovary. From the ovary, what is going to be produced? From the ovary, ovum is going to be produced. So over here I'll just write ovary. Cool. Now, talking about this particular thing that you see, this is known as what? The fallopian tube. So what we do, we write it over here. This is known as the fallopian tube. What is the other name of fallopian tube? Guys, what is the other name of fallopian tube? Oviduct. Exactly. It is also known as oviduct. Oviduct. Manoj is saying, ma'am, if at all you had completed second chapter, I was scoring out of out. Oh my god, I'm so sorry, dear. But I really hope you had watched the one shot. Anyways, this is also known as oviduct out there. Now we go ahead and talk about each parts of the oviduct. Okay, the portion that we have right over here, this is known as the finger-like projections, and you have a name for it. What do you call the finger-like projections as? You call this as fimbriae. You call this as fimbriae. And this fimbriae is a part of what? It is a part of something that is known as infundibulum. Okay, infundibulum is the first part of your oviduct, and it has finger-like projection, which is known as fimbriae. Then you have something like this, which is known as ampulla. It is known as ampulla, where the ovum is going to come and stay, and this is where, okay, this is going to be where the fertilization is finally going to happen. Finally, you have one more thing right over here. This is going to be known as the isthmus. 3 portions makes up the oviduct out there. Now, at the top of the uterus, you make something like this and write uterine fundus. This region is known as the uterine fundus. And finally, you can connect this line at here. You can connect this line also over here. And you can mention the outermost layer of the uterus. Guys, uterus has 4 walls. Check. Uterus has 3 walls. Can you name all of it? Syed, blueprint of UT1. Blueprint of UT1, this is almost the blueprint. Obviously, I will be solving a lot of question out there, but yes, this is definitely going to come. I cannot say that this question will definitely come because there are only few and the syllabus is very vast, right? But yes, I'll definitely try. Guys, I'm asking you something. Okay, Pavan says endometrium, myometrium and perimetrium, ma'am. Exactly. So in the uterus, we have 3 walls out there. Okay, 3 walls. The outermost wall which is there, it is known as perimetrium. Okay, it is going to be a thin wall, that is going to be a protective wall. Then we have a middle wall over here. What do you call it? You call this as myometrium. Myometrium is made up of what, my dear children? Anyone can guess? Myometrium is made up of what? Perimetrium is going to be thin wall, and this thin wall is there for what? Protection. This thin wall is there for protection. I am asking you, champions, what is myometrium made up of? Any guesses? Come on. Myometrium is made up of what? Ma'am, I also wanted to say one more thing. I have seen a lot of biology teachers, whether online or in my college, but most of them are just irritated most of the time. But you are very humble, ma'am. Thank you so much, Pavan. That's so sweet of you. Guys, I'm waiting for you. Myometrium? No one? No one knows what is myometrium made up of? So I'm going to go ahead. Myometrium is made up of a muscular layer. It is basically a muscular layer. It is made up of smooth muscles. And you know, smooth muscles are not voluntary muscles. Basically, you cannot control them. For example, I have smooth muscles over here. Inside my, basically, what do you call that? Alimentary canal is made up of smooth muscles, which is why the food that I eat basically goes down. There is contraction, there is relaxation, and I cannot control it. Basically, this thing, if at all I am doing something like this, what are these muscles? These are skeletal muscles. This is under my control. So over here, when I talk about myometrium, it is made up of smooth muscles. It is not under our control at all. And finally, you have an innermost layer. What do you call this as? This is known as endometrium. This is known as endometrium, and endometrium is going to be a glandular layer. This is the only layer which is there. This is the only layer which is there, which is going to undergo cyclic changes. Means every time it is going to shut down, again form, again shut down, again form. This is a diagram of the female reproductive system that you have to make. Is this clear? Do you have any doubts around this particular diagram that you want to ask me? You can ask me right now. Ma'am, is there any chapter-wise weightage for this test? No, Macha. Syed, there is no chapter-wise weightage. Mostly, like, you will be getting a mix of questions from almost all the important chapters out there. Okay, all these chapters that you see, principles of inheritance or human reproduction or sexual reproduction, they are all important. Little less important is reproductive health, but that's a very small chapter. So, now, no doubt, ma'am, it is very, very clear. Okay. Ma'am, I'll meet you in the next live class, ma'am. Now, I have to prepare for maths tomorrow. Manoj, wishing you the best, go rock. Okay, so yes, now I'm going to go ahead with the next question which is there, and that is draw a neatly labeled diagram of the sectional view of the mammary gland. This is another important diagram that could be asked to you. Can I go ahead with making this diagram, or if you feel that diagram practice is already done, I'm going to skip this. See you too, Manoj. Broody, ma'am, I'm not able to download my NEET application slip through my KA. My class is C. Dear, I have to check it. I cannot give you verbal confirmation over here. I'll definitely try to find a solution for it, okay? And I'll let you know. Okay, guys, do I need to go ahead, make the mammary gland, or can I skip it? Will you practice by yourself? Come, tell me. Then only we can go ahead. We have so many questions. Akash, Pavan, waiting for your answer. Okay, then probably I'll make it, since no one over here is talking about it, then I have to make it. Okay, Then, okay, yes ma'am, we can skip it. So one person said we can skip it. Okay, in that case I'll skip it. Listen to me, this particular diagram is important. It can come over here. What do you have to, oh, okay, one very important thing. Sorry Macha, I cannot skip it. I have to give you a very important trick. So this is a usual question that is being asked, that is, please tell the flow of the mammary gland. Okay, the flow of the mammary gland, and I'll tell you a way to remember the flow of it. It's very, very important, class. Okay, and it goes like this. Dal fell on mat. What is it? It is like dal fell on mat. Okay, and the trick goes like this. So first of all, we have to write mat, and then we have to write dal. Then we have to write dal. Over here, M stands for mammary lobes. Over here, M stands for mammary lobes. How many mammary lobes do we have? Almost 15 to 20 mammary lobes are there. Okay, there are almost 15 to 20 mammary lobes. Inside each lobe, what do you have, Baba? You have so many alveoli. You are going to have so many alveoli. These alveoli which is there, it is going to open into what, Baba? It is going to open into tubules. Okay, they have tubules. Tubules which are there, they are going to join together to form what? They join together for forming duct. Duct together comes together to form what? They're going to form ampulla. And the ampulla which is there finally forms together what? The lactiferous ducts. It is finally going to form what? The lactiferous ducts. So always remember dal. Okay, dal which was there, it fell on what? It fell on mat, and this is the flow that you have to go. This is very, very, very important. Okay, you can get an MCQ based question on this, and also it is important in terms of marking in the labeling as well. As I said, this is a 5-marker question that I am discussing, the sectional view of the mammary gland. So lobes, alveoli, alveolar sacs, tubules, ducts, ampulla, lactiferous duct, and then the nipple. Exactly. Over here, one more very important thing. A is right over here and A is over here also. How do you remember whether it's alveoli or ampulla coming first? Listen, ampulla ends with la, and la is what? Lactiferous duct. This is how you can go. Shantali, this is, Baba, this is the important questions that I am discussing over here. All the questions that I am talking about is important. When you learn the concept, you will be able to solve the MCQs as well. That is why I came up with direct conceptual based questions. Okay, so that I can explain you things. We'll be solving some MCQs and we'll see whether we are able to solve it or not. Okay, but first concepts. Cool. Now if you talk about the diagram could be simple. First of all, you have to make something like this. Just a second. Okay, so you can make something like this, give an opening over here and make a line like this over here. This is your ribs. Okay, over here what do you have? These are your ribs, and between that you have the muscles. So these are your ribs and that is the muscle. Then you have to make fat. You know, the breast would be having so much amount of fat. So fat will be there all throughout. Now what we can do is, the easiest way to draw this particular diagram is to go ahead and make a lot of alveoli. You have to make what? A lot of alveoli. If at all you are able to make alveoli up to this particular point, it will be easy for you. So you make something like this over here all throughout. Okay. Similarly here also, you're going to draw this particular diagram like this. This is going to be what? This is going to be alveoli. Mention that this is going to be alveoli. Now after alveoli, what do we have? First of all, we have lobes. So basically in this we have multiple lobes, which I'm just making over here. I had made only few of them because in your NCERT also it's somewhat looks like that. Further, what do we do? This alveoli are going to open into what? Tubules. So you have to make tubules. So make a tubule over here. Come on, my pen does not work. Okay, so over here there is a tubule, there is another tubule. Okay, similarly make few tubules over here also. Here also you can make tubule, and you can just join it up. Listen to me. These tubules which are there, they're going to come together to form what? Duct. Similarly, these tubules come together to join what? To form what? Duct. Here this also come together to join what? Duct. So this is how we are going to make it. Finally, we have the ducts. These are going to be ducts. So first of all, mention that they are what? Tubules. After tubules comes together, they form what? Duct. Now the duct which are there, they are also going to come together. Okay, the ducts which are there, they are also going to come together. What do you label that as? You're going to label that as the next thing, which is ampulla, and finally we show that all the ampulla are going to open up at this particular point like this. This is going to be the lactiferous duct, and finally this particular region which is there is going to be the nipple region. Okay, you do not have to make a complicated diagram out there. It's a very easy diagram. Just make T branches. You just have to make sure from the point you have alveoli, you have these many branches. You have these many branches to make. Okay, that's a basic logic that you have to use. Even in the diagram that they had given, this looks like this. You cannot find clear segregation of things. Okay, so this is your alveoli. Then you have your tubules which are hidden. Finally, they have mentioned duct. Okay, they have directly mentioned duct. The ducts which are there, this is a duct, this is a duct, this is a duct. They all come together to open what? Into ampulla. The ampullas, they come together to form what? Lactiferous duct. That is what they have mentioned. Similarly, we also have to go, as I said, the diagram that I have made would be more than enough for you. You do not have to do too much of creativity out there. Labeling has to be done in parts. You can also mention as 1, 2, 3. That will be way more easy for the teacher to give you marks. Mention mammary law first, then the ampulla, then the tubules, then the duct and everything. And as I said, you can use this formula for mentioning that. Dal fell on mat. If it's clear, can you give me thumbs up, dear children? If this particular concept is clear, can you give me a thumbs up? Ma'am, you are taking both paid batches and YouTube classes, right? Yes, dear. Okay, guys. Okay, ma'am, aren't nipple and the lactiferous duct same, ma'am? Because NCERT have changed, the milk is sucked from the lactiferous duct to nipple this year. So basically, lactiferous ducts opens into nipple. That is, nipple is basically a region. It's not a duct, it's basically a region. You call this as nipple region. That's all. So this is clear, right? So I've got a thumbs up, so I believe it's clear. Next is schematic representation of spermatogenesis. Guys, from here things are going to change up. We are going to actively involve. I am going to ask you things and you are going to tell me the answers for that. Okay, schematic representation of spermatogenesis. If at all this is being asked, what is the first cell that you have over here? What is going to be the first cell that is going to be over here? Any idea? From which point onwards the spermatogenesis is going to begin? What do you call the first cell which is going to be deployed in nature? If you are asked, spermatogonia, exactly. So apart from Pawan, anyone else also want to give the answers for this? Baba, go ahead and give me the answer. First cell that we have is spermatogonia, okay? Now the spermatogonia says that I want to divide, I want to increase my number. So it is going to undergo what division? It is going to undergo what division? Is it, okay, is it, this thing that is mitosis, or it is going to undergo meiosis? Remember over here, it just want to increase the number, okay, it does not want to do anything else. The number of chromosome is going to remain the same. Which division will it undergo? Will it undergo mitosis or meiosis? So Pawan says it is mitosis. Guys, anyone wants to try it up? Ma'am, reply correction window. Dear, I have to check. Shreya. Sorry. Okay. Inchara is also there. Good evening, dear. Okay, so this particular division, yes, it is also known as equational division. By the way, this is what we have. This is known as mitosis. Okay, we have mitosis, we have increased the cell number. Now listen to me. From this point onwards, things are going to change. What do I mean by that? Once the cell divides into two, one cell says that I would be like my father. I am going to be like my father. I am going to make sure that the cell keeps on dividing. The other cell says that I don't want to be like you. I want to become this sperm. I am going to become this sperm. This particular cell which is there, which decides to form sperm, first of all you need to understand this cell is going to undergo some transformation. It will undergo some transformation as in maturation, no division and all. You call this process as differentiation. By differentiation I mean that there are going to be some changes happening within the cell. They are going to get matured and finally they are going to give the matured cell. What do you call this? Yes, Pawan has told, it is known as the primary spermatocyte. You call this as the primary spermatocyte. Okay, the primary spermatocyte. Now from this primary spermatocytes, we are going to go ahead with the actual process of, division to form the sperm. Until now, we were just doing the background thing. Okay. Okay, Shreya ma'am, please make a video on correction window will open or not. Shreya, I'm really unaware of that particular thing at this particular point of time. The reason being that I'm talking about KA medical. Okay, in that case, you're talking about option entry. Is it that you want to fill in the option, or you want to correct any of your details? If you want to correct any of your details at this particular point of time, I do not have any information regarding it because they have not updated it. If at all they do so, I'll definitely make a video on it. If you're talking about option entry, you want to make a new list adding the medical seats, the round 2 option entry has to begin and it will take some time. Okay? Okay, now I'm going to go ahead. Okay, so spermatocyte is right over here. This primary spermatocyte that we have, Baba, is going to undergo division. How does it undergo division? So over here you have the primary spermatocyte. It is now going to undergo which division? To form sperms, you're talking about gamete. Okay, you are talking about gamete. So formation of gametes requires what? Which division? Meiosis. Exactly. So over here meiosis is going to happen. The cell is going to divide, but you are going to see some unique thing. The number of chromosome which was deployed first, okay, we had 46 number of chromosome, it becomes half of it, it becomes haploid. What do you call this cell as? If this was primary spermatocyte, what do you call this cell? What do you call this cell? This is known as secondary spermatocyte. Exactly. It's known as the secondary spermatocyte. Okay, it is known as the secondary spermatocyte. Now what happens? The secondary spermatocyte again is going to divide. Why divide? Because it has meiosis, and meiosis happens in 2 stages. So this is the first stage that we have. This is going to be the second stage, Baba. We will be having one more stage. At the end of it, what do you get? The secondary spermatocyte, when it divides, what do you get? Do you directly get the sperms, or you get something else? Do you directly get the sperm or something else? Meiosis to give rise to 4 equal haploid spermatids. Exactly. So listen to me carefully. Primary spermatocyte divided to form secondary spermatocyte. Secondary spermatocyte over here, Baba, are going to give rise to 4 cells. This cell divided into 2, this cell divided into 2. The number of chromosome, will it reduce now? It has already reduced in the first division. Will it reduce now? The answer is no, it is not going to reduce. It is going to be remaining the same. It remains the same, Baba. What do you call them as? They are known as spermatids. They are known as spermatids, and this is very, very important for you to know. Baba, the spermatids which are formed over here, okay, I want to ask you a question. Are they the actual sperms? Are they the actual sperms, or you need to make the sperm now? What do you think? Are they the actual sperms, or you need to make the sperm now? These spermatids undergo differentiation to give rise to sperm through the process of spermiogenesis. Exactly. So listen to me carefully. These guys which are made, the spermatids which are made, no Baba, over here, they are globular structure. Okay, they are round globular structure. The sperm is not like that. Sperm is elongated structure. So if at all you want to convert them, you need to do something about it. Okay, and this is not going to be a division process. The cells are not going to divide further. Rather, they are going to mature and they are going to differentiate. They are going to differentiate. So this is again a differentiation process that we have, a maturation process. But finally, you are going to get your sperms like this. What do you call them as? You call them as spermatozoa. You call this as spermatozoa. Okay, you call this as spermatozoa, and listen to me. This process of formation of spermatids to spermatozoa, this conversion is known as spermiogenesis. And for those of you who usually get confused, let me tell you, sperm already always wanted to make itself. It was not able to make itself. Right now it says that I want to make me. Spermiogenesis, let me be me. Okay, spermiogenesis is the process that we have. Always remember, spermatogenesis is the whole process that is there. Finally, when the sperms are being made right over here, they are going to say, Tata Baba, I have been made over here. I am going to move out of the seminiferous tubule. What do you call that process as? Again, all the things that I am saying could be coming as MCQ, could be coming in any manner that is there. That is why I am explaining in depth. Okay, when I say that these sperms, spermiogenesis happen and finally spermatozoa are there, these spermatozoa are going to move out. Okay, are going to move out to the seminiferous tubule. What do you call that process? Anyone who is watching this video, can you tell me the name of the process, Baba? And Shreya, as soon as I find your answer, Baba, I will be replying to you. As of now, there's a class ongoing, which is why I'm not able to pay attention to all the things that is there. But I'll try my best to find a solution for it, okay? Guys, what do you call that process? of release of sperm, that is spermatozoa, from the seminiferous tubule, it is known as, any idea? It is known as spermiation. Spermiation, always remember aviation, like that you can remember spermiation. So spermatogenesis is there, here we have spermiogenesis. Who converts into what? Easy thing. Spermatids, they are like kids, small chutku kids. They are immature, they are globular in shape, and finally they differentiate, they mature into spermatozoa. This spermatozoa says tata bye-bye to the seminiferous tubules and move out. This is known as spermiation. All this process is important. If at all you write all of it very nicely, you're going to get your whole 5 marks. Or this time in your FA-1, they probably can be giving you this and can be asking you associated questions out there. So probably you should be knowing about all of it. Am I clear with this? If I'm clear with this, can you guys give me a thumbs up? If Baba, I am clear with this thing, can you give me a thumbs up? Yes, ma'am. Perfect. Let's go ahead then. Next question that we have is, okay, so we have already made this. Draw a label diagrammatic sectional view of the male reproductive system and label the parts: testis, seminal vesicle, glans, penis, epididymis and urethra. Okay, this is again a diagram based question which is there, which is again important. How do you make this diagram? How do you remember things? Before that, okay, just give me a second. Okay, are you? Can I go ahead and make this diagram? Will it work if I try to make this diagram over here right now? If yes, can you give me a thumbs up? Human male reproductive system, again a 5-marker question. That is why I'm putting efforts in making the diagram, otherwise I would have skipped it. Okay, so it goes like this. Listen to me very carefully again. Over here you have the testis. Okay, this is the testis that is there. In the testis, Baba, in the testis you have so many. How many testicular lobules are there? Any guesses? How many testicular lobules are there? Almost 250 testicular lobules are there. Okay, testicular lobules, TL, team lead is what I remember. Inside each testicular lobules, you have almost 1 to 3 seminiferous tubules. You have almost 1 to 3 seminiferous tubules. So you make that 1 to 3 seminiferous tubules. Where are these seminiferous tubules opening into? Okay, in that case, makes sense. So I'm assuming, children, so Pawan said that, ma'am, we can leave the drawing diagrams, right, because we can see and understand them in NCERT. In that case, if you're pretty sure about the diagram, I'll leave the diagram part and we'll move on to the theory, because I also know that it takes a lot of time. Okay, anyways, so in that case, let's go over here, let's talk about this. Okay, so here is a question that is basically a 5-marker question, and it goes like this. Given below is a flow chart showing ovarian changes during the menstrual cycle. Fill in the spaces with hormonal factors responsible for the events shown. Okay, so over here it is a menstrual cycle flow, the ovarian changes in the menstrual cycle, and you have been asked this particular question, that is basically primary follicle is there, primary follicle goes into mature graphene follicle. What is A? Okay, then graphene follicle gives ova. What is B? Then ova basically leads to corpus luteum, and over here you have C. Okay, do you know the answer for this, or shall I go ahead and explain the menstrual cycle? Can I go ahead and explain the menstrual cycle? It's totally fine, Pawan, no issues. That makes sense. Guys, anyone know the answer for this particular question? What is the one that converts primary follicle to graphene follicle to ova, and basically corpus luteum over here is going to release what? That is known as C. That is the question. Okay, to solve this question, we have to know about the whole menstrual cycle, which is exactly why I am going to go ahead with it. Make sure you listen to me very carefully, because this is a very important concept. No, ma'am, not very sure about the answer. So listen to me very carefully. Okay, in the menstrual cycle, if at all we see, suppose this is a cycle that we have over here, we have few phases. Day 1, suppose we talk about day 1. Day 1 to day 5, which is there, we say we have a phase that is known as menstrual phase. And I am believing every question that comes related to this topic is going to be clear. Menstrual phase is there. Okay, from day 1 to day 5, menstrual phase is there, where what is going to happen? The endometrium which is there, it is going to shed down. Shedding down of endometrium. Why do you think the endometrium is going to shed down? So always listen to me very carefully. You have two sets of hormone. One set of hormone is known as ovarian hormone. Okay, these are released by the ovaries, and basically under that comes what? Under that comes estrogen, and the next one which is there is known as progesterone. Okay, so you have estrogen, and the next one that we have is progesterone. They are what? Ovarian hormones. They are ovarian hormones. Similarly, you have pituitary hormones. These hormones basically are going to be released from the pituitary gland. They are going to be released from the pituitary gland. Which part of the pituitary? Baba, it is going to be anterior part of, so pituitary gland which is there, no? It has anterior lobe and the posterior lobe. So anterior lobe of the pituitary is going to release two hormones. Very, very important hormones. They are known as FSH, and it is going to be known as LH, follicle stimulating hormone and luteinizing hormone. Now here is the trick. Listen to me. These guys are enemies. They do not like each other at all. Estrogen, progesterone does not like FSH and LH. That is a basic logic. Then you need very importantly these particular hormones, that is estrogen, progesterone, for the maintenance of endometrium. If at all you want to maintain the endometrium, then you need estrogen and progesterone. Now when I say that day 1 to day 5, the endometrium is shedding down, what do you mean by that? When I say that the endometrium is shedding down, what do you mean by that? That basically these two hormones are in very less amount. Estrogen and progesterone are going to be in very less amount. Also, I do understand one more very important thing. When these guys are in less amount, who increases their amount? Basically is the other guy which is there, which is FSH and LH. They say that my Shatru is not there, my enemy is not there. This is the correct time for me to basically increase my amount. So over here at this particular point, what is going to happen? You will be seeing an increase in FSH as well as LH. FSH and LH are slowly going to increase. This was the menstrual phase from 1 to 5 days. Until now, is it clear? If clear, can you give me a thumbs up? If it is clear, can you give me a thumbs up? Menstrual phase, is it clear? Estrogen, progesterone is less, FSH, LH is increasing. Okay, slowly they are increasing, and here you see endometrial breakdown. Why? Because these two guys are less. Okay, got it. Yes, ma'am. Now what happens? FSH, while it increases, it also makes sure about one very important thing, that is FSH is follicle stimulating hormone. It is going to stimulate the follicles. From the primary follicles in the ovary, they are going to now convert into what? The secondary follicles. The secondary follicles which are there, they are now going to convert into what? The tertiary follicles. That is going to take place over here. Under the influence of what? Under the influence of these two hormones, that is basically the FSH and LH, but majorly the FSH. The follicle stimulating hormone, under the influence of that, this is going to increase. Which is why from day 5 to the next few days out there, we say we have entered the new phase. What do you call this new phase as? This phase is known as follicular phase. What do you call this phase as? Just give me a second. You call this phase as the follicular phase. Why do you call this follicular phase? Because Baba, over here the follicles are developing. That's not the only reason. There is one more reason. Okay, that's okay. Follicular phase. Okay, Pawan is saying follicular phase or proliferative phase. Exactly. So this is the one name, that is follicular phase. You also call this as proliferative phase. Why is the reason for that? What is the reason for that? So basically, while the FSH is increasing and it is making the follicles to grow, the follicles over here are going to be backstabbing the FSH. Okay, they are going to backstab the FSH by releasing one very important thing, that is known as estrogen. And estrogen is what? FSH Shatru, that is the enemy. But estrogen has now been released by what? The cells of the follicles, that is the granular cells of the follicles. They are going to release the estrogen. Now you only think, if estrogen is being released over here at this particular point, don't you think that is going to help in the formation of endometrium? Yes. Slowly over here, the endometrium is going to get reformed. Under the influence of estrogen, I am telling you, slowly the endometrium is now going to get reformed. Okay, now that is happening. Now, what is the thing? FSH is there, follicles are generating, the follicles are releasing what? Estrogen. When estrogen is now being released, do you think FSH can go on increasing? No, their enemy has come, they cannot move forward. FSH cannot keep on going, increasing, rather its amount decreases. And why is it important? Because when I say the follicles are developing, Baba, there are so many primary follicles, they're generating into secondary, they're generating into tertiary, but not everyone can form graphene follicle. We cannot let that happen. Not everyone should be forming graphene follicle. So you should know FSH level should go down. Definitely it should go down. Okay, so under the influence of estrogen, FSH goes down, LH also goes down. And while this is happening, anyway the follicles, they are going down, they are not nil, they are there. So follicles are developing, they are still releasing estrogen. There comes a time where the level of estrogen is going to go up really high, really high. And at this time it says that you are my enemies, but you also, Macha, you also be happy, do not be worried. FSH, LH, all of them, they increase in amount. So I'm telling you, there comes a time over here, almost on the 14th day, where what is going to happen? The level of estrogen is so much. Okay, so much. At this time, it says that enemies, you also be happy. LH, FSH, you also increase in your amount. This leads to something which is known as LH surge. It leads to something that is known as LH surge. And on the 14th day, Baba, what is going to happen? This graphene follicle which was being formed over here, it is going to rupture. Under the influence of LH, LH goes and acts on the graphene follicle, and basically it ruptures and releases what? What do you have inside the graphene follicle? You have the secondary oocyte. Secondary oocyte is now going to be released. That is the logic. Yes, exactly. It is known as the ovulation day. It is going to be known as the ovulation day. So you have your graphene follicle. I'm just making it over here. This is the graphene follicle. It has now released what? The secondary oocyte over here is now going to be released. This is known as what? Ovulation. So ovulation happens on the 14th day. It happens, done, dusted. Is it clear? And by the way, since endometrium is reforming under the influence of estrogen, slowly you also call this phase as proliferative phase. Because why? Because the endometrium is proliferating. That is why you also call this as proliferative phase or ovulatory phase. I hope this is clear until this particular point. Until the 14th day, everything is super clear. Right, Macha? Guys, you are free to ask me doubts. You are very much free to ask me doubts if you have any related to these topics. It is very, it will be really good if at all you ask me doubts, because in that way it will be more interactive. Yes, ma'am. Okay, until this particular point it is clear. Now we go ahead. What is going to happen? Your graphene follicle has ruptured, secondary oocyte is released, secondary oocyte goes where? Into the fallopian tube. Until now everything was happening inside the ovary. Now the secondary oocyte goes to the fallopian tube and waits for the sperm to come such that fertilization happens. It goes to the ampullary region and waits there. Okay, while it happens, the endometrium says, or the uterus says, that I have to be prepared. Anytime fertilization can happen and basically a zygote can be formed, embryo will be formed. I need to nourish it. I have to make myself very, very thick. The endometrium has to be thickened. So what do I do is that basically with the leftover thing that is there in the graphene follicle, now I'm going to rename it into something else. This particular thing that we have left back, it has a name. You call this as the corpus luteum. You call this as the corpus luteum. And corpus luteum is here because of what? Because of LH. Always remember. LH surge caused the rupture. So which is why I'll say LH is the reason why we have corpus luteum right now. And the corpus luteum at this particular point of time, Baba, is going to release two very, very important things. It is going to release two very important things. One is going to be your progesterone, and the other one that we have is going to be your estrogen. Progesterone and estrogen is going to be released. Okay, ma'am, done. These two things are released. What's the use of these two things? Baba, estrogen was already making the endometrium. When progesterone also comes into picture, it is going to thicken the endometrium. If at all the endometrium was like this, it will thicken the endometrium way too much. And not just that, it will make the endometrium start secreting certain things, start secreting certain important nutrients out there. Why? Because the endometrium is thinking that one day embryo will come to me, okay, and I have to provide nourishment for this particular thing. That is why the endometrium, that is why, what do you have? You have basically okay, wait. So Ankit sir has come and I forgot everything. Okay, just give me a second. Where was I? He's saying endo. Wait, no. Yeah, he was, yeah. So basically embryo could be coming and I could be providing nourishment to it. That's the basic logic, okay? So what you are doing is that under the influence of progesterone and estrogen, you are making the endometrium secrete things, which is why you say that this is the phase which is also known as secretory phase. You also call this phase as secretory phase. And not just that, since corpus luteum is doing all of these things and LH, luteinizing hormone, is taking care of it, I'll also call this phase as what? Luteal phase. So I have luteal phase and I have secretory phase is nothing, the same thing itself. It's this phase that is from the 14th day and it goes up to what stage? It goes up to the 28th day. Now here, just imagine, okay, just imagine fertilization has not taken place. Okay, the sperm and the egg has not come together, zygote is not there, embryo is not being formed. What is going to happen? If embryo is not being formed over here, there is a twist that is going to happen in this particular story. Progesterone says that I waited for a very long time, I cannot see this, embryo is not there, I prepared everything and now until now embryo has not come and I'm really unhappy that LH is there. LH is my Shatru. How can it be there? I do not want LH to be there. So it sends signal. Progesterone which is there, it sends signals to our brain, actually hypothalamus, and in the hypothalamus basically it is going to send signal to our pituitary gland and say that hey LH, you have to be stopped. Now if LH is not there, do you think there is someone to maintain this corpus luteum? No. Corpus luteum is not going to be maintained. If corpus luteum is not there, progesterone is not going to be there. If progesterone and estrogen is not there, do you think that over here your endometrium is going to be maintained? No, Baba. What is going to happen over here? The shedding of the endometrium happens over here. That is exactly why I said E and P goes less. Why? Because fertilization did not take place. Hello Manju. Am I clear with this particular thing? This is known as luteal phase. This is known as secretory phase as well. Cool. Is it clear? Can we go ahead and solve the question now? Can we go ahead and solve the question now? So yes ma'am, it is very much clear. Perfect. Cool. Now I'm going to go back and we're going to reach up over here. Where is it? Yes. Now you tell me the answer for this. Okay, primary follicle is there and you have to make the Graafian follicle. What is A that is going to help the primary follicle convert into the Graafian follicle? Manju and Pavan and everyone who is watching this video, now you can try this particular thing. Come on children, you can do it. Primary follicle to Graafian follicle. Which particular hormone is responsible for this particular conversion? Follicle regeneration. It has a term follicle in its name. It has a term follicle in its name. Come on, guess Madi. Waiting. No trying, only waiting. Okay, so over here, this particular hormone that we have for the generation of primary follicle to this Graafian follicle is known as follicle stimulating hormone, FSH. You know it is the only one that is responsible for generation of the follicles. So it is going to be FSH. Always remember that. Now this FSH is going to make the Graafian follicle. From the Graafian follicle, we need ova. We are going to need ova. How do we do that? Graafian follicle is there and we need ova. For that, ova basically means the next part which is there. Manju is saying FSH now. Okay dear, thank you so much. Now I am asking the second part, B part. From Graafian follicle you need the ova. Which hormone helps in that? LH, exactly. Luteinizing hormone. Manju, perfect macha. Okay, it is going to be the luteinizing hormone which is going to help over here in basically rupturing of what? The Graafian follicle. Okay, rupturing of the Graafian follicle and the release of what? Basically the ova. I hope that is clear. Perfect. Now the ova basically later on, so over here they have formed corpus luteum directly. So you feel that ova formed the corpus luteum? No, basically that's Graafian follicle that forms the corpus luteum. From the corpus luteum, what is being released? What is going to be released, Baba? That is C. Whatever the corpus luteum is going to be releasing, that is known as C over here. So what does it release? Okay, my actual name is Dhanu. Okay, I'll try to call you Dhanu itself, dear. Last question. What does the corpus luteum release over here? Come on. Progesterone, exactly. Progesterone or estrogen, either way, that will be the last thing which is there, and you have solved the answer. That is basically. So first one we have FSH, stimulates the development of primary follicle to the graphene follicle. Second was luteinizing hormone. The LH surge causes ovulation, that is release of ovum, actually secondary oocyte. And finally, progesterone secreted by the corpus luteum maintains the endometrium for the possible implantation. Cool. This is how it goes. Now you have the B part of the question that says women experience two major events in their lifetime, one at menarche and the second at menopause. Mention the characteristics of both the events. So what do you mean by menarche? Menarche is basically the start of the reproductive cycle. The first onset of menstruation at puberty. It marks the beginning of the reproductive phase in the female, that is known as menarche. What do you mean by menopause? It is the permanent cessation, that is ending of the menstruation. It marks the end of the reproductive cycle. Can you tell me, yes Pawan, start and the end of the menstrual cycle. Can you tell me what is the age by when someone gets menopause? By when someone has menopause? Can you guess that particular thing? Age. Almost at 50 years, exactly. Almost at 50 years there will be menopause. For Manju says 45 to 50. Perfect. Oh, good girls. Good girl and good boy out there. Perfect. Pawan ma'am, can I ask you not read it? I will ask after this chapter. Yes, sure, Pawan. You can. After this chapter, you can ask me. Cool. This question is super clear. We're going to go ahead with the next question. This is a 5-marker question. Now the next question says, draw a sectional view of seminiferous tubule of human and label the following cells. So basically when you are asked the sectional view of seminiferous tubules, you have to draw this particular diagram. But before drawing this diagram, let me talk about this question. Okay, so seminiferous tubule, you're talking about spermatogenesis as a whole, and here are some questions. In spermatogenesis, cells that divide by mitosis to increase their number, what are these cells known as? Cells that divide by mitosis to increase their number, just increase their number. What do you call it? You call them as spermatogonia. Remember, we spoke about it. Spermatogonia is there. It increases the number by division. That is known as mitosis. So this is going to be spermatogonia. Perfect. Power. Next one. Cells that undergo meiosis 1. What is the first one to undergo meiosis 1? You have got spermatogonia. It underwent mitosis. Now you had got two cells. One cell divided again. The other cell said that I will form sperm. I will get differentiated. So that cell get differentiated. Okay. And it formed the primary spermatocyte. The primary spermatocyte forms what, Baba? Okay, Pawan says primary spermatocyte is the answer for this. Actually, the cells that undergo meiosis 1 is going to be the primary spermatocyte. Okay, now cells that undergo meiosis 2. Guys, cells that undergo meiosis 2. Come on. This is the primary spermatocyte. Okay, this is 2N. It underwent meiosis 1. You got two cells that is going to have half the number of chromosomes. What is this cell that will undergo meiosis 2? That is the question. What is this cell that undergoes meiosis 2? Okay, secondary spermatocyte, is that the answer? Exactly. Primary spermatocyte forms the secondary spermatocyte. And also one very important thing that you need to remember over here, for the first time it is going to become haploid. The first cells to become haploid over here is going to be secondary spermatocyte. Exactly, Pawan as well as Dhanu. The next question is there, cells that help in the process of spermiogenesis. Cells that help in the process of spermiogenesis. What do you mean by spermiogenesis? Over here you get what? You get spermatids. They are globular. You want them to be converted into sperm, that is spermatozoa. For this you need a process that is known as spermiogenesis. Spermiogenesis. You have been asked this question, cells that help in the process of spermiogenesis. Any idea? Spermatid. No, that's not the answer. That's not the answer. It's not the spermatid that will be helping in the process of spermiogenesis. Okay, I'll just, what do you say? I'll just pause this question over here. I'll ask the next part of the question, okay? Cells produced due to spermiogenesis. What are the cells that are produced due to spermiogenesis? Can you try that? What is the end result of spermiogenesis? What is the end product of spermiogenesis? That means when I say that, basically here I got spermatid. Okay, here I got spermatid. Then I told you these globular cells convert into something. Okay, convert into something. What do you call them as? Basically, this process is known as spermiogenesis. This process is known as spermiogenesis. And my question is, what do you call these cells as? Spermatozoa. Exactly. You call them as spermatozoa. If at all you ever get confused between this and this, as in who comes first, I'm telling you again, spermatids, they are like kids, small kids, they are globular, okay, and they have to mature to form spermatozoa. This is known as spermiogenesis. Okay, Pavan says D is FSH, right? No, FSH is a hormone. D over here, the question is cells that help in the process of spermiogenesis. This is the question. Cells that cell, not a hormone. So since everything else is being sorted over here, by the way, I have written everything answer. So over here, the answer for this question, the D1 is going to be Sertoli cells. Is going to be Sertoli cells. And if at all you have a confusion regarding that, I am going to quickly move into the next, that is again anyway the 3-marker questions. Over here, I have a 3-marker question that will be giving you the answer for this also. So basically, there are some hormones which are super important in the whole process of spermatogenesis. Okay, and these hormones are released from so many people out there that you see over here. Okay, and this is a 3-marker question also. Study the following flow chart, name the hormones involved at each stage, explain their functions. So while I give you the answer for this, you will be able to understand that why is the answer for this question Sertoli cell. Cells that help in the process of spermiogenesis, why they are known as Sertoli cells, why are they not any other cells out there. So listen to me very carefully. It's a very easy thing. Okay, it's not at all tricky. First of all, we have the grandfather. Okay, first of all, we have the grandfather that is known as hypothalamus. This is where, this is there in our brain. Okay, the hypothalamus which is there, it is going to release a hormone, gonadotropin releasing hormone. What does it release? Gonadotropin releasing hormone. Basically, it's a command that this particular hypothalamus is giving to someone else. Who is it commanding to? It is commanding to the next gland out there, that is known as pituitary gland. Okay, so basically they cannot talk to each other, so that is why they use chemical messengers that are hormones. So over here, gonadotropin releasing hormones goes and act on the pituitary gland and says that, Macha, you have to release some hormones. Now the pituitary gland listens this and says that, okay sir, I am going to release some hormones. Over here you have got 2 hormones that they release. One is going to be LH, luteinizing hormone. Other one which is there is going to be FSH. Always remember, pituitary gland releases these 2 hormones. LH and FSH cannot be released by anyone else in the entire body, only pituitary gland. And they are actually gonadotropins. They act on the gonad, that is basically testis and the ovary. They are known as the gonads. So that is why it is written, gonadotropin releasing hormones act on pituitary glands, and finally gonadotropins are being released. Cool. Until this particular point, is it clear? Until this particular point, is it clear? If yes, can you give me a quick thumbs up? A quick thumbs up, line by line. Children, why I'm asking you to give me a quick thumbs up is that's when I realize that you are understanding things. If you have any doubt, you have to ask me. Yes, ma'am. Okay, perfect. So Bhavan has understood. Dhanu, I'm hoping that you have understood. Kantesh is also there. Hello. Srijan, okay, PDF LA sigute, you will be getting the PDF in the WhatsApp channel. You can join the WhatsApp channel, and also there is an all-in-one page over here. There also you'll get the PDF, but as of now in the WhatsApp channel. Okay, perfect. Thank you. And Mahantesh is there. Perfect. Now everyone has come. Okay, now going ahead, what happens ahead is that this LH which is there and this FSH which is there, they are going to do something. They are going to do something where inside these testes. Inside the testes you have testicular tubules. Inside that you have seminiferous tubule. Suppose this is one seminiferous tubule. This is another seminiferous tubule. Here you have 2 cells within the seminiferous tubule. One is going to be the Sertoli cell. Other one which is going to be there is known as the spermatogonia. This spermatogonia is later going to form the sperms, and who provides nourishment? Sertoli cells, right? Okay. Now here in between the seminiferous tubules you have space where you have some cells. What do you call these cells as? Leydig cells. What do you call them? Leydig cells. So Leydig cells are also there. What does the Leydig cell do? If at all you remember, they are responsible for the production of hormone. What hormone? The male sex hormone, that is androgens. You call them as what? Androgens. Now the most famous androgen that we have was what? what? Testosterone. That is testosterone. That is the male sex hormone that we have. That is a major one, is testosterone. That is the basic logic, right? Right. Now listen to me. The LH which is there, it goes and acts on the Leydig cell. Okay, it goes and acts on the Leydig cell, LL, you can remember like that, and says that, hey Macha, I have been getting orders from hypothalamus. You have to release testosterone. You have to release the male sex hormones, basically testosterone or anything that is there. Okay, that is happening where? Inside the testis. Do remember that. Okay, so this guy releases testosterone. FSH on the other hand says that I also want to go and act on something. So FSH goes and acts on what? The Sertoli cells. Sertoli cells here, right? It goes and acts on Sertoli cells and says, hey Sertoli cells, you release some factors. You release some very important factors. What are these factors? They are actually androgen binding factors. The name of this is not given in NCERT, I'm telling you. They are known as androgen binding factors. What is the androgen? Over here, testosterone is the androgen. So to bind the androgen, to make sure that the testosterone is being maintained in a high level, these factors are being released. Okay, now I'm going to show you a picture over here. This is your testis where the sperm formation is happening. Where the sperm formation is happening, this is spermatogenesis that is taking place, right? So what is happening is that testosterone, which is released over here under the influence of LH, which is there. Now the testosterone moves inside where? Inside the seminiferous tubule. So basically I can say it has come up over here for the whole process to happen. Now for the whole process to happen, you have to make sure the level of the testosterone is maintained really high. So that is why you need the factors to make sure that Macha, please do not go anywhere. You be here only. I'll take care of you. Okay, that process is also taking place very fine. Now I am asked that one question, that is, what influences spermiogenesis? That means over here you have got the spermatids. Okay, they are globular in shape. You want to form the sperms, that is spermatozoa. There is a process that is known as spermiogenesis. Baba, over here, the cell is not dividing, the cell has to differentiate, it has to mature, it has to undergo some changes in the, structure, where you need high amount of, where you need the help of Sertoli cells. Sertoli cells promise that I am going to nourish you every now, every then, right? Every now, every then. So these Sertoli cells have to be in super high amount over here for the process of spermiogenesis to happen. So ma'am, for the whole process they are not needed. For the whole process they are needed. They are releasing factors. The factors are super important for maintaining testosterone. That and all is there. But over here, when I say differentiation is happening, some cytoplasmic changes is happening, I really need Sertoli cells over here for the process of spermiogenesis. That is why in the previous question, if at all you remember, cells that help in the process of spermiogenesis, the answer for that question was Sertoli cells, because they are the one who promise that I am going to nourish you. I am going to nourish you. So that is the basic logic. And always remember, in your NCERT, there is a dialogue that is given that if FSH is not produced, which process is going to be affected the most? The most affected process which is going to be there is spermiogenesis. Spermiogenesis is the most affected process. If at all you have been getting options like that, it will be spermiogenesis. Am I clear with this? Mom, oh, I like the way how you're teaching the classes, man. Thank you so much, Pavan. Guys, am I clear with the whole thing? This is very important thing. Clear? If clear, give me a thumbs up. If clear, give me a thumbs up. Okay, Mahantesh and yes, perfect. Thank you, Srujan, Srujan, Pavan, and where is Kantesh and Dhanu was also there. Anyways, so this is how it goes. Now coming back to the question, study the following flowchart, name the hormones involved at each stage and explain their functions. Tell me, hypothalamus, what do they release? What do they release? Hypothalamus. What is the hormone that they are going to release over here? And basically, the hypothalamus is going to be actively releasing these hormones when we hit puberty. Okay, when we hit puberty, that's when they are going to release, and that's when the whole process is going to happen. Guys, I'm asking you a question. Hypothalamus is going to be releasing what? Which hormone? Gonadotropin releasing hormone. Exactly, it is going to release GnRH. Okay, now pituitary gland. What does pituitary gland release? What are the hormones that the pituitary gland release? Come on. They're also going to release some hormones, right? Pituitary gland, what do they release? LH and FSH, exactly. Luteinizing hormones and follicle stimulating hormone. Testis, what do they release? Testis, what are they going to release? And other children, everyone who is watching this video, Macha, listen, I'm not here to judge you. I'll never do that. Please make sure you try to answer even if it's wrong. Try to answer. If you get it wrong, probably I'll tell you other way to get things correct. That's the easiest way, right? N2O. Sorry. Oh, androgen is what you meant. Okay, Mande says androgen. Yes, exactly. Testis is going to release androgen. That is, and the most famous androgen that we have is testosterone. Now, finally, we have these sperms. Now, finally, we have got these sperms. So, for the formation of sperms, which hormone is required? For the formation of sperms, that is, for the formation of spermatozoa, which hormone is highly required? For the formation of spermatozoa, which hormone is highly required? The last one. Come on. And then we are done with this question. For the formation of the sperms, or basically spermatozoa, you need a hormone specifically, without which spermiogenesis cannot take place. What is that name? What is the name of that particular hormone? Guesses, guesses, guesses. Final one. It is going to be known as okay, it is going to be known as your basically, where is it? And the outcome, the formation of the maturation of sperm, follicle stimulating hormone. Exactly, it is going to be FSH, which is going to later give rise to the sperm out there. So basically, you have been asked about hormones involved in E stages, and I hope now it's basically FSH only. It's not wrong. Follicle stimulating hormone. Okay, so this question is super clear to all of you. Will you be able to write it? Hypothalamus, GnRH, pituitary, FSH and LH, testis, testosterone promotes spermatogenesis and development of male secondary sexual characters, and finally outcome is the formation of maturation of sperm. It has been influenced by what? It is influenced by FSH. Okay? You, wait a second. Come on, work, Macha. Yes. Perfect. Cool then. Let's go ahead with the next question. Name the pituitary and testicular hormones involved in spermatogenesis. Come on, Macha, do it. Now, since you have anyway understood everything, okay, let's go with pituitary first. Name the pituitary hormone. This is again a question that had come. Name the pituitary hormone involved in spermatogenesis. Come on, children, everyone. I want equal participation, okay? Try. Pituitary hormone. An anterior pituitary gland is releasing this. Always remember. What is the pituitary hormone that helps in spermatogenesis? Mahantesh, Dhanu, Srujan, Pavan, LH and FSH. Exactly. LH and FSH from the pituitary. And what do you have in testicular hormones? You have basically all the androgens. If at all you want to specify one, you will say testosterone. You will say testosterone. It will be better if at all you write androgen and in bracket you write testosterone. That will be the safest one. Cool. And yes, Pavan has given me, Mahantesh has also given me the answer. Perfect. Perfect. Now, we are going, by the way, this is how we have to frame the answer. Pituitary hormone, FSH, what does it do? You have to mention that. Secreted by the anterior pituitary, acts on the Sertoli cells of seminiferous tubules and stimulates the whole process of spermatogenesis and also spermiogenesis. Okay, so while writing the answer, do not show any conjugacy. Please make sure you write enough answer. LH is also known as this. You do not have to write that. Okay, you can just say secreted by anterior pituitary, acts on the Leydig cells and helps in the secretion of testosterone. Testicular hormone, testosterone, secreted by the Leydig cells. What do? They are essential for the maintenance of the whole process of spermatogenesis. And then it is done. Now we come to the next question, that is, what is menstrual cycle? Mention one pituitary and one ovarian hormone that plays a role in the menstrual cycle. So we discussed about the menstrual cycle, the entire reproductive cycle that you have. This is known as menstrual cycle. It is of 28 to 29 days out there. Okay, and there are definitely pituitary hormones and ovarian hormones. Can you name the pituitary and ovarian hormones? Let's go with ovarian first. Can you tell me the name of two ovarian hormones that we had discussed? So are these PUC and NEET similar, ma'am? Because I can answer all the questions and we see this type of questions NEET, but mainly in objective format. Yes, the concept anyway is going to be similar, which is why you find all of the similar things. Okay, got it. Shankar, 5-marker questions could be coming from principles of inheritance and variation. Again, this particular chapter, that is human reproduction and sexual reproduction in flowering plants. All of these 3 chapters give 5-marker questions usually. Okay. Okay, Shobha says estrogen and progesterone, exactly, and basically Pawan also says estrogen and progesterone. Ovarian hormones are 2, there is estrogen and basically we have progesterone. Finally, we have pituitary hormones also. What are the pituitary hormones? We all know it. It is going to be FSH and LH. We discussed about it, FSH and LH. So that is also done and dusted. Mohantesh also gave me the answer. So how do you frame the answer? Menstrual cycle is what? Is a monthly cyclic change in the ovary and the uterus that occurs in the human female for about 28 days. FSH stimulates the growth and maturation of the ovarian follicles and also induces them to secrete estrogen, right? When the follicles are developing, they are releasing estrogen. So you have to mention that. Now estrogen, what does it do? It causes repair and proliferation of the endometrium, and apart from that, if you remember, if at all, okay, if at all, Baba, estrogen goes up really high, actually they are shatru enemies, but estrogen when it goes up really high says that enemy, you also be happy, there will be LH surge, and LH surge leads to what? Ovulation. Okay, cool, works fine. Now going ahead with the next question out there, what is placenta and list its function? Come on, this is going to be new for you guys. What is placenta and list its function? Can you try? What is placenta and list its function? Okay, tell me the function of it. Placenta is, I am telling you what is placenta. Placenta is going to be a temporary vascular tissue that is going to be formed between the fetus as well as the mother, okay, which is going to help with 4 very important things. Can you tell me the functions of placenta? Can you tell me the functions of placenta? And I'll tell you how it is being formed. Okay, I'll start with how it is being formed, then you can probably come up with the functions of the placenta. Okay, placenta is a type of endocrine tissue formed between fetus and mother during pregnancy. Okay, you can also mention it is a vascular tissue which is filled with blood vessels, which is being formed between the fetus as well as mother during pregnancy, and I'm going to tell you how it is being formed. So Baba, listen to me very carefully, okay? You had the zygote, single cell. Okay, this is single cell. This zygote divided into 2 cells, 2 into 4. I do not have 4 hands, otherwise I would have showed you. 4 into 8. That is how it goes up. This is known as cleavage to the uterus. Basically, we say the process is cleavage. But the zygote, which is single cell, okay, it is basically dividing to form 2 cells, 2 to 4, and it goes on like that. There comes a stage where it forms almost an embryo. Which how many cells? There will be almost 8 cells to 16 cells out there. This particular stage is known as morula. This is known as morula. This is a stage that we have in the fallopian tube itself. Okay, until the fallopian tube, it's there. So Raju is saying removal of carbon dioxide. Yes, Macha, I'm coming to that. You're correct. Okay, so this is morula. After morula, Baba, after lots of division, there comes a stage. There comes a very important stage over here. You call this stage as, you have to tell me the name of the stage, okay? You guys have to tell me the name of the stage where there is outer layer of cells which you call as trophoblast, and there will be inner layer of cells which you are going to call as inner cell mass. You will call this as inner cell mass. Now I am asking you one major thing, that is, what do you call this particular stage as, where you have an outer layer of cells and when you have inner layer of cells? What do you call this as? Gastrolayer. No. Try. Shobha, once more. This stage is very important. Okay. This stage is very important where it goes and get implanted into the uterus. In this stage, it goes and gets implanted into the uterus. For example, I'll say suppose this is the uterus. Okay. And here you have your endometrium. In this stage, it comes inside the uterus and basically gets implanted like this. It is going to get implanted like this. What do you call this stage as? Blastocyst, ma'am. Exactly, Shobha, exactly, Pawan. It is going to be known as blastocyst stage. This is very, very important in terms of MCQs and everything. In the blastocyst stage, it comes and gets implanted over here. The outer layer is known as trophoblast. The inner thing is known as inner cell mass. Now what happens? Now there are going to be some changes. When this guy comes for the first time, you know what is going to do? The trophoblast which is there, it says that I am a part of embryo, but I am going to do something very important for you. I am going to take part in the formation of placenta. I will be helping in the formation of placenta from the embryo side. Similarly, from the uterine side, there will be blood-filled spaces and things like that, which is going to be contributing for the formation of placenta. Okay, so I'm telling you one thing. From the embryo side, the trophoblast, not the inner cell mass, the trophoblast helps in the formation of placenta. From the mother's side, the endometrial tissue, or basically the uterine tissue, helps in the formation of the placenta. How does it happen? Look at this very carefully. The trophoblast which is there, it is going to make finger-like projections like this. It will be making finger-like projections like this. Inside this, what do we have? We have our inner cell mass. Inside this, Baba, we would be having our inner cell mass just like that. Just the trophoblast goes and make finger-like projections. What do you call this finger-like projections as? You call this finger-like projections as chorionic villi. You call this finger-like projections as chorionic villi. So when it's asking what is morula, Baba, morula is a stage. When I say the zygote is a single cell, it divided, you got a stage of embryo where almost 8 to 16 cells are there in the embryo. This stage of an embryo is known as morula. Okay, the blastocyst, you have no mention about how many cells and everything will be there. That is not given in NCERT, but about blastocyst they have mentioned one thing, that basically this is the stage where it comes to the uterus and gets implanted. Okay, now I'm telling you the trophoblast which is there, it forms a finger-like projection which you call as chorionic villi. This is from the embryo's end, and from the mother's end, in between these spaces there will be blood-filled spaces, and together they are going to contribute some maternal tissue as well. Later on, this whole structure which you see over here is going to look different. This inner cell mass is later on going to form the whole fetus out there. What happens to this? This basically forms the placenta, and both of them are going to be connected with the help of one very important thing which is known as the umbilical cord. It is going to be known as what? The umbilical cord. So basically, can I say that trophoblast helps in the formation of placenta, because trophoblast helped in the formation of what? Chorionic villi first, that later on merged with the uterine tissue. They both together form the placenta. And what happens to the inner cell mass? Inner cell mass later on forms the embryo proper. That means trophoblast was a martyr. It said that I am not going to be known as a part of the embryo anymore, or basically I am going to form the placenta. You become the embryo, the actual embryo, which will later divide to form the fetus. That is the basic logic. Am I clear with this? Ma'am, what is implantation? Implantation means basically, suppose here you have a embryo. This embryo has to go and get embedded, get inside the endometrium, has to take ultimate support from the endometrium. For example, I am there. Okay, I go and get inside this particular screen. Okay, literally inside, so that I get all the nourishment from the screen. This is known as implantation. Got it? Clear, Macha? Fine. So chorionic villi is also explained. The reason I'm talking about this is because the next question you are going to see chorionic villi, how it is going to be formed. So morula 8 to 16 cell, these cells are known as blastomeres. Okay, all the cells which are there, they are known as blastomeres. Later on, blastomeres, the cells are known as blastomeres. So we say 8 to 16 blastomeres. Here you have so many blastomeres, the number of cells is not being mentioned. Okay, anyways, now I'm going to come to the question, that is placenta. Placenta is a vascular connection. Vascular means there will be so many blood vessels and stuff like that. Connection between the mother and the fetus that helps in nutrition, respiration, excretion and hormone secretion. How nutrition? Okay, how nutrition? So in nutrition, basically it transfers nutrients from the mother to the fetus. Papa, basically the baby which is there, it cannot have food. Okay, so whatever food is there in the mother's blood, whatever important nutrients are there, that is going to be diffused. Baby does not have it, the mother has it. So from higher concentration, things are going to go to the lower concentration. That is basically within the fetus body. Got it? So that is how it helps in nutrition. How does it help in respiration? Similarly, the baby which is there, it is not over here, it cannot breathe air and things like that. So mother's blood brings oxygen. Okay, now oxygen is high inside mother's blood but less inside the fetus. From higher area it will come to the lower region. Okay, and the baby anyway, since it is getting oxygen, it is going to do respiration, Baba. And when it does respiration, what is the end product of respiration? When you do respiration, you get CO2. This CO2, if it is accumulated, we will die. The CO2 has to move out. So same thing happens. The CO2 will move from the fetus this time to the mother. From the fetus, it moves to the mother. That is how it helps in respiration. Then we have excretion. Again, in the baby's body, there will be so many metabolic waste because respiration, a lot of things are happening. This has to move out. So excretion also will be happening through the fetal blood itself. And finally, this is the most important function of the placenta. That is, it acts as a temporary endocrine system. It is going to act like a temporary endocrine system. What do you mean by that? Basically, why do you call it temporary? Because only during pregnancy you see placenta. Right, and I am saying it is going to release some hormones, which is why I'll say that it has an endocrine function. Endocrine glands means the ones that has the potential to release the hormones. Now, I am going to ask you a question. What are the 4 hormones that are released by the placenta? What are the 4 hormones that are released by the placenta? Come on, try it up. Ma'am, you love teaching, right? Because I can see that happiness in your eyes, right? Oh my God. Yes, I do love teaching, and more than that, I love teaching you children. Okay, so when you communicate with me, it becomes all the more easy for me to, enjoy this whole process. Now, ma'am, yava chapter in the 3 marks question, but ma'am, please answer. Okay, so in FA-1, that is not specified here, but in general, I think it's this chapter, that is human reproduction, from where you get 3 marker questions, reproductive health, from where you get 3 marker questions, and also sexual reproduction, flowering plants. Okay, but in FA-1 it's not been specified like that, to be honest. In midterms you will see that. Okay, cool. Guys, I'm asking you a question. What are the name of the hormones? Come on. What are the name of the hormones that is released by the placenta? Everyone, what are the name of the hormones that are released by the placenta? I'm waiting for you guys. Raju, and where is Shankar, Mahantesh, Shobha, Pavan, everyone, try Madi. Great thing about religion, always teach while smiling. Oh my God, thank you so much. HCG, HPL, estrogen and progesterone. Is it? What about others? Others also have the same answer? So if you do not remember, always, it's very important to know placenta, which is a temporary endocrine gland, releases 4 things. Raju says HCG. Perfect. HCG stands for what? Human chorionic gonadotropin. Okay, it is known as human chorionic gonadotropin. Later on, the next thing that we have is HPL, human placental lactogen. HPL is going to act on the breast, okay, that is the lactating membrane of the female, and it will help in the production of the milk in the female, okay. The next one that we have is progesterone. And the last one that we have is the estrogen. Now I have a question. Progesterone and estrogen were released by ovaries, no ma'am. How can placenta do this? Baba, during pregnancy, placenta says that, Macha, you ovary, you rest, Macha. I am going to take care of it. I will be releasing progesterone and estrogen. Okay, so always remember this logic. Do not forget it. Placenta releases 4 hormones. HCG, human chorionic gonadotropin. What does the HCG do? So if you remember, if at all you remember, there was corpus luteum. Right, corpus luteum under the influence of LH was releasing progesterone, estrogen, and the progesterone, while it was there, it didn't like LH, and it said that I do not want LH to be there, let LH go down. When LH was going down, basically corpus luteum was going down and progesterone was slowly dying. It did not know that I am doing a bad thing for myself. Right, now when fertilization happens, you need endometrium, that means you need progesterone, but progesterone does not like LH. So in what way can you maintain the endometrium? There's only one way, that is, instead of LH, find someone else who can maintain the corpus luteum. So always remember, when someone gets pregnant, you have the trophoblast. That trophoblast, which was forming the placenta, also helps in releasing of some HCG. It is helping in the formation of placenta. It has not formed the placenta yet, right? Placenta formation takes a lot of time, almost 3 months. Okay, so until that time, this particular role is done by HCG for maintenance of corpus luteum, such that progesterone and estrogen can be released, such that the endometrium can be maintained. We need it, which is very, very important. Which is why the usual in pregnancy, the test which is done, it is urine test or any test in general. The level of HCG is checked. If the level of HCG is more, that means that person is going to be pregnant. Okay, now when the placenta fully forms, it takes up this duty, and it has HCG, HPL, progesterone, estrogen. Cool. Okay, so now coming to that relaxin part, listen to me. So this is about placenta. They release 4 hormones. Now I'm asking you another question. During pregnancy, okay, there are 3 hormones which are going to be increased in the mother's blood. It is already there in the mother's body, but during pregnancy they are going to be increased in the mother's blood. Can you tell me the name of those hormones? Can you tell me the name of those hormones? Come on. During pregnancy, these hormones are going to be increased in the mother's blood. Name of these hormones? Ma'am, during pregnancy of other hormones. Raju, I didn't get your question, dear. During pregnancy, in the mother, there are 3 other hormones which are going to get increased. They're already there, they will just increase. Come on, come on, come on. One starts with T, I'll tell you, okay? I'll give you a clue. In the mother's blood, increases in many folds. You have 3 of them. One starts with T, one starts with C, another starts with P. Any guesses? Any guesses? What are these? Anyone can guess it. Waiting, waiting, waiting. Prolactin. Okay, perfect. Who said that? Who said that? Pavan said that. Okay, we have prolactin over here. P stands for prolactin. Perfect. Prolactin is another hormone. As I said, it goes and acts on the breast and basically helps in the production of milk. Okay, prolactin is increased. Second thing, okay, T for thyroxine. Exactly, T for thyroxine. Thyroxine is something that controls all our metabolic rate and everything. For example, for making of things, for breaking down of things, we need thyroxine hormone. Okay, so that is thyroxine. And you are not sure about C. Anyone, can you guess what is the C one which is going to increase in the mother's blood during the process of pregnancy? Any idea? Any idea that you have? Okay, I'm gonna break that part then, since no one has the idea. It is known as cortisol. It is known as cortisol. Cortisol is also responsible for the metabolism, fetal metabolism, and also the mother's metabolic rate and things like that. So thyroxine, cortisol, and prolactin is there, which is going to increase in the mother's blood during the pregnancy. It is already there in the body, but it's just going to increase. Placenta, on the other hand, releases these 4 hormones, and now we just have one guy. Okay, and we just have one guy out there now that is left behind. You remember placenta told ovary that macha ovary, you relax macha ovary, you just relax out there. So ovary took it very seriously and it started releasing relaxin. It started releasing relaxin. But this relaxin hormone is going to be released during later phase of pregnancy. Not in the initial stage, but it is released during the later phase of pregnancy. Always remember this. So we have first this, the hormones of placenta. Second this, the mother's blood, what are the hormones that are there, and then we have this, that is ovary releasing relaxin, but during later phase of pregnancy. And I'll tell you a secret that I think Pavan already spoke about, and that is, my dear children. Okay, Raju is saying thyroxine and cortisol. Yes, dear, perfect. Okay, so one secret is that actually placenta is also releasing relaxin in the initial phases. Okay, why do we need relaxin? First of all, for making the ligaments and the pelvic ligaments and everything relax a bit, such that the baby can easily move out during the process of parturition. That is the logic. Okay, so over here relaxin is released by the placenta as well, but in your NCERT it is not been mentioned, which is why you do not need to be worried, but know that. Okay, so if in a question, if at all you are getting that what are the hormones released by this particular thing, what do you say, placenta, HCG is given, HPL is given, and then you have FSH, and then you have relaxin. If you are being asked which hormone is not released by placenta, what will be your answer? I'm asking you a question. If you are asked which hormone is not released by the placenta, which one will be your answer among this? Guys, try Madi. Which one is not going to be released by placenta? That is the question, okay? HCG, HPL, relaxin, or FSH? Pavan, Raju, Mahantesh, Sudha, everyone. Can I tell then? Can I tell then? Bolo ma'am. Okay, too much bolenge. The answer for this question is going to be FSH. Listen, relaxin, I told it's released by the placenta also, but actually in your NCRT, according to your NCRT, it is going to be ovary who is going to do the function. But when you see FSH, FSH cannot be released by anyone apart from pituitary. So that guy has to be moved out. Okay, that guy has to be moved out for sure. So this one would be the answer. Okay, in that case, if at all that comes, but in generally these type of questions would be avoided for you in second PUC. It could come in your KCET and all, but yes, generally FSH, exactly, Raju. Cool. So these are the few things that I wanted to tell you, and now I'm going to go ahead with the next question, and that is, parturition is induced by complex neuroendocrine mechanism. Comment. What will you say? What will you put down as an answer for this particular question? Parturition means what? Expulsion of the baby. The baby is fully formed. Baba, now it wants to move out of the body. Okay, it wants to move out of the body, and I am saying it is done by complex neuroendocrine mechanism. How do you explain this? Neuroendocrine means nervous system is involved, endocrine system is also involved. How do I explain this particular answer? Can I go ahead? Can I explain parturition to you guys? If yes, can you give me a thumbs up? If yes, can you give me a thumbs up? Raju, I think your answers are coming up a bit late. It could be a lag in the network or things like that. Children, I'm asking you a question. Shall I explain parturition to you? If yes, give me a thumbs up. Fast. Okay, if no thumbs up, that means no explanation is required, no? Okay, thumbs up. Okay, Mehrun Mustafa is saying, okay, thumbs up. Listen to me. So over here, you have the uterus. Where is it? Where is our uterus? Yes. I'm going to shift the uterus somewhere over here. Cool. I'm so sorry. Yes. Now listen to me. The baby is right over here. Okay. The baby is here. The baby is attached to the placenta right over here. And over here, what do you have? You have the cervix. This is going to be the cervix, and this is going to be the vagina out there. I'm just writing over here, cervix. Cool. This is the uterus, this is the placenta, and over here, the innermost lining that you have in the placenta is going to be the endometrium. Endometrium. Cool. And over here, below that, you have another layer which is super important. What do you call this as? You call this as myometrium. You call this as myometrium. What is it? It is going to be the muscular layer. Okay? This muscular layer which is there, it is made up of smooth muscles. That means that they do not listen to anyone but only the brain. It is involuntary. It is not under our control. Okay, fine. Now here starts. And one more thing. Here there are so many receptors. Okay, what do you call these receptors as? Stretch receptors. They are known as stretch receptors. Okay, if at all there is a stretch, they are going to send signal. Now comes the story. Baby says, I want to move down. Okay, I want to move. I am fully formed. I just want to see the world. In this particular condition, when the baby is fully formed, the fetus is fully formed, it is going to release some hormones. It is going to release some hormones over here. These hormones are going to communicate to the placenta, and now placenta is going to release two hormones. You do not have to remember the name of these hormones. I'm just telling you the story so that it will be easy for you. They release estrogen and they release prostaglandins. Okay, they release estrogen and prostaglandins. Okay, now listen to me. Estrogen, which is released from the placenta, okay, by the information given by the fetus, makes some receptors over here super active. What are these receptors and where are they getting active? They are getting active in the myometrium. What do you call these receptors as? They are known as oxytocin receptors. Okay, they are known as oxytocin receptor. So there is oxytocin, which is a hormone, and this is the receptor for it. This is done by estrogen. That is done. Okay, done, understood. Let it be there. What does the prostaglandin do? Prostaglandin says that, Macha, you want to move out? No, I am going to cause some contraction over here. Okay, I am going to cause some contraction over here. So this leads to contraction. Now you only imagine. Here contraction is happening. Here again contraction is happening. Mild uterine contraction is going to happen. Slowly the baby is going to move down. The baby is moving down. down, while the baby moves down, what happens? The stretch receptors over here are going to get active. They're going to say emergency, emergency. Now this particular stretch receptors are going to send what? A nervous signal, or basically I can say neuronal signal to what? To the brain. Whose brain? Mother's brain, obviously. Mother's brain. In the mother's brain, what do you have? You have the hypothalamus. Apart from it, you have one very important thing, that is the pituitary gland. The pituitary gland. Which type of pituitary? You have anterior and posterior pituitary, no? This time it's the posterior pituitary. Always remember. Parturition, posterior pituitary, okay? Which now releases a hormone that is known as oxytocin. Oxytocin. Now this hormone is released, this hormone is carried through the blood, it comes over here, it comes and binds on the receptors. Who made the receptors already? Estrogen made the receptors already. Here you have all the receptors onto which what is coming and binding? Oxytocin is going to come and bind. When the oxytocin going to come and bind on the muscular layer, what is going to happen? Contraction, relaxation, contraction, relaxation. And when that happens, the baby will be moving down. Again the stretch receptor is active. Again it sends the signal, again the same thing, repeats until the baby and the placenta and the umbilical cord is totally outside. So first of all, let me ask you a question. Who started this off? It is basically the fully developed fetus as well as placenta that started this off. This is known as the fetal ejection reflex. The first reflex that you have is known as the fetal ejection reflex. It is the one that causes mild uterine contraction that starts off the process. Okay, then we say that this contraction leads to the movement of the baby and starts and basically activates the stretch receptors, which sends nervous signals to what? Pituitary gland. Now the pituitary gland releases oxytocin. From here it becomes hormonal. This hormonal signal comes and acts on the uterus and causes complete contraction and relaxation. And finally, what is going to be happening? The baby will be moving out. How do you frame this answer? Parturition is brought about by a complex neuroendocrine mechanism involving both the nervous system and the endocrine system. Both of them are involved over here. Second thing, at the end of pregnancy, the fully developed fetus and the placenta initiate mild uterine contraction, which you call as what? Fetal ejection reflex. You do not mention about prostaglandins, estrogen and things like that. Finally, as the baby's head stretches the cervix, the stretch receptors in the cervix send nervous signals to what? Hypothalamus, which stimulates the posterior pituitary to release oxytocin. Oxytocin causes stronger uterine contraction, which stretches the cervix further, creating a positive feedback loop, and finally the baby is going to be out of the body. This is a 3-mark question that have come from this chapter. I hope you have understood it. Is it clear? Is it clear to all of you? If yes, give me a thumbs up. If you have doubt, ask me right away. And by the way, every one of you is watching this video, you might be seeing a thing that is given over here. Join the free NEET KCET 2027 course. If you click on that particular link, basically you will be moving on to a page where you will be asked to buy something. But when you click on buy, it will be 0 rupees. You do not have to pay any fee. It's actually a structured way of providing you all the lectures that are done in YouTube, because you know there are so many counseling videos and things like that. So if you're told you want to find subject wise all the videos which are there, and also some assignments are yet to be uploaded, but yes, structured videos are definitely there. You can check it out, and that too for free. It's all in one page. Okay, so go make sure you check it out, and you do not have to give much of information, just phone number is there, so it will be really good if at all you go ahead and do so. Good, let's go ahead now. So Raju gave me a thumbs up, and I hope Pawan and everyone have understood this question, Baba. Okay, I believe I can go ahead. Okay, just give me a second. Ma'am, next question. Perfect. I think, okay. When and where do chorionic villi appear in humans? State their function. So chorionic villi, we discussed about it. Chorionic villi appear after implantation, after it comes and gets implanted. Okay, during the 2nd week of pregnancy, they develop from the trophoblast of the blastocyst. They develop from the trophoblast layer, the outermost layer, yeah, and project in the maternal endometrium. Chorionic villi together with maternal uterine tissue forms the placenta, increasing the surface area for exchange of gases and things like that. Function, transfer of oxygen. Same same function. Basically, the function is to form the placenta. And what is the other function of placenta? You just mentioned that, and it will be done and dusted. Okay, so that is the role of chorionic villi. Less chances for this question to come straight forward like that, but yes. Okay, finally we have one more part and then we are done. Okay, then we can move on to the next chapter. So most important topics apart from these things that we have discussed so far is basically month by month development data. What happens in each month? So listen to me very carefully. We'll just talk about it and then it's done. So in your NCERT, 6 months, what do you say? What is happening in all the 6 months? That has been mentioned. Nothing beyond that. And if you know the answer, you have to tell me. Okay, I'm going to ask you questions. The first month, second month, third month, then fourth month is skip, but anyway, fifth month and then sixth month. Can you tell me what happens at the end of the first month? Can you tell me what happens at the end of the first month? First month. When? Okay, if at all you do not remember the things as such, I'm going to explain. Listen to me very carefully, okay? By the end of the first month, when someone dies, what do you check for the first? Check heartbeat, right? So by the end, when someone is born, that is the first thing that is going to be formed. You will be having heart formation. You will be having heart formation by the end of first month. And the first heart sound. Exactly, Raju. Perfect, dear. Now, what happens in the second month? That's an easy way to remember it. You form the heart like this, right? So I can say the second month, the limbs, that is this, and the digits are being formed. The limbs and basically the digits are being formed by the end of the second month. Limbs and the digits. Now you only imagine over here, there is a baby, okay, with heart and limbs and the digits out there. What else do you need? You will obviously need a lot of things over here inside the body. You need organ and everything out there. So I can say second fetus. Second fetus. So I'm talking about fetus only. This is fetal development. Okay, when a baby is being formed like a baby, I'll say fetus especially. So it is heart, then we form limbs and the digits. Okay, limbs as in hand and the legs and also the digits. Finally, for filling in all the spaces that we have almost all the organs as well as the organ system that is going to be formed. Organs and the organ systems are going to be formed, and also one more very important thing happens, that is the external genital organ is being made over here. The external genital organ is going to be made over here. So three things happen: organs, organ system, and the external genital organ is also going to be made over here by the end of the 3 months. Am I clear on this until this particular point? Okay, heart, limbs and digits, and then to fill the space, organ system and the external genital organ, that is penis in the case of males, and in the case of females, mons pubis and what do you say, mons pubis, and the other part, what do you call this as? Oh my god, I forgot the name. Come on, remember. Labia majora, labia minora, and all those things. Cool? Now, next thing that happens in the fourth month, it's skip, because that is not given in NCERT. So we'll skip it. Okay, and we'll directly come to the fifth month. Fifth month is interesting. Fifth month, the baby says that, God, until now I listened everything to you. I'm actually getting very, very bored inside. I want to move around. I need to have hair on my head also. So the first movement, always remember, fifth month, So first movement is going to be seen over here, and apart from that, hair on the head. Hair on the head. First time you will be seeing hair on the head of the fetus in this particular month. Now God said that you wanted hair. No, have hair all over your body. Baby asked for just this thing, hair on the head. The God said that have hair all over your body. So in the 6th month you will be having hair on the body, and apart from that, eyelashes are going to be formed. Okay, eyelashes are going to be formed. Someone said eyelids. Yes, and eyelids which are there, eyelids are going to get separated. Okay, they are going to get separated, as in basically these eyelids are going to get separated. That is thing that is going to happen. 6th month, that is eyelids. Yes, perfect. And Meher and Mustafa, perfect, dear. Cool. These are the things that is there. Please remember it. First month heart, second month limbs and digits. The third month you have organ and organ system and also the external genitalia. Fourth month skip madi. Fifth month what do you have? First movement and also hair on the head. 6th month hair all over the body, and finally eyelashes will also be there. Eyelids are going to be separated. One more important information. 1 month means how many weeks? 1 month basically means 4 weeks. So question can be coming like this. What happens in the end of 4 weeks? So you should say heart is being formed. What happens in the end of 8 weeks? 2 into 4. 2 months meet 8 weeks. What happens at the end of 8 weeks? What happens at the end of 12 weeks? So you should know it's 3 months. Okay. What happens at the end of 20 weeks? 5 into 4. What happens in the end of 24 weeks? 6 into 4. So that is the logic. By that they would be asking you a question. The first 3 months are known as first trimester. The last 3 months will be known as the second trimester. Cool? Yes, motivational. Yes, in some cases the parturition is in early stage also. That can happen. Cool, ma'am, I'm not able to download my UG NEET application slip after entering the NEET roll number in KCET. Dear, I would have to check, Saroja Shetty. I do not know about it. I would really have to check. Okay. So guys, I hope you have understood it. Why, ma'am? It can be premature development. That could be, let's say, really some fluctuations in the hormones and things like that, which is why prostaglandins is already active, and it can lead to mild contraction, things like that. Okay? Yes. So, children, can I go ahead with the next thing? If it's clear. Akash, where were you? I have completed one chapter. Okay. So then I'll go ahead with the next thing that is there. So yes, and I want to tell that you children are rocking. Now I really want to ask you a question. Will you be going ahead with solving the MCQs which are there over here? Shall I go with the long questions for the other chapters? Can I go ahead with the long questions for the other chapters? Okay, you have, I think, 2 chapters, Reproductive Health and Principles of Inheritance and Variation. I was planning to go ahead with Principles of Inheritance and Variation because I think that's an important chapter. Shall we? Yes, right? So sorry. By the way, everyone, make sure you drink water and keep yourself also hydrated. Cool, then I have to move to Principles of Inheritance. By the way, tomorrow you will get the Reproductive Health chapters one shot. Okay, you are going to get the one shot for the Reproductive Health chapter. So every question is being covered up there also. So if you wish, you can watch up. It's not like there's something that is left empty. Okay, if you wish, you can go ahead. Come on. Now we had this first question out there. Schematic representation of one gene inheritance in the case of monohybrids. So monohybrid means one gene. We're talking about only one gene and the cross of it. So listen to me very carefully. When you talk about one gene inheritance, you're talking about only one character there. Okay, you are talking about just one character at that particular time. This character, for example, if I talk about height, height is a character. Under height comes traits. Under height you have trait. Okay, and you have 2 trait out there. Okay, you will be having 2 trait. So under height you have trait, one is tall and the other one is going to be what, Baba? Dwarf. Other one is going to be dwarf. And for everyone who probably have no idea how genetics and everything works, listen to me very, very carefully. Okay, so when I say that a gamete is being formed, so over here when I say 46 number of chromosomes are there, and in another guy, so this is your female and this is your male. When I say 46 number of chromosomes are here and here, and when I say the gametes are being formed, I say only 23 chromosomes are going to go into each gametes. By that I mean one very important thing. That means that if at all you had 2 chromosomes over here, a pair of chromosomes over here, basically only 1 chromosome is going to go into one gamete. From one pair, only 1 is going to come. So this is 2. Out of that, only 1 is going to come. That is why I'm saying when 46 chromosomes are there, out of that only 23 are going to come. And in each chromosomes that you see over here, Baba, you are going to have something. So for example, I talk about this. These are chromosomes, right? And in the chromosomes, you have some segments which you call as genes. They are nothing but the DNA only. And the segments of those DNA which what do you call this as? You call this as genes. Now I am saying that this particular gene is controlling height, this gene is also controlling height. Okay, and I am saying that this gene is capital T, here also this gene is capital T. What do you mean by that? That this gene is responsible for height, which is tall. How it is responsible? Basically, it is responsible for making up a protein which is super important for giving us good height out there. Okay, that is a basic logic, or basically this is forming a protein that will be helping in some ways to give us a good amount of height out there. Similarly, in some cases what is going to happen? In some cases these are genes for height only. One can be capital T, another can be something else. It can be small t. What do you mean by that? This gene is for height only, but this gene is not going to produce enough amount of proteins such that I will be having a good height, such that I am going to have a good height. So this type of logics could be happening, and always remember, from a pair of chromosome, only one is going to come down into a gamete. Only one is going to go into a gamete. Okay, so this is basic logic. If at all you have understood the basic logic, you will be able to go ahead with all of these things. So here when I say about one gene inheritance, I am talking just about height, and I am saying, when I am to show this, I am saying that I am crossing two parents. One parent which is going to be tall, just a second, one parent which is going to be tall, and the other parent which is going to be dwarf. And I also mentioned a term before this. I say that they are pure breed. Pure breed tall. Here also it is what? Pure breed dwarf. What do I mean by that? I mean that here both the chromosomes are going to be same, having the same gene out there, capital T, capital T. When I say pure breed dwarf, it means both the chromosomes have the same gene, that is small t, small t. Got it? This is what? This is parents. So you have to mention parents. Then you have to go ahead and make the gametes. Now in the gametes, I told you that from each pair only one chromosome can come into a gamete. So over here, from this pair one chromosome comes. Suppose this is a gamete, it will be having one chromosome. Anyway, whatever you do, the only thing that it can contribute is capital D gene, because both the chromosomes have the same only. Similarly over here also, you get small t in one, another small t in another gamete out there. Now this is going to be the first generation, filial one generation. When I cross this, I'm going to get the first generation out there. What do I get in the first generation? Listen to me very carefully. Suppose there is a male gamete carrying capital T chromosome, and there is a female gamete carrying small t chromosome. When both of them come together, you get capital T, small t. Okay, you get capital small t. What happens in the next one? Let's calculate. So this went with this. Now suppose this went with this one. Again I get capital T small t. Suppose this went with this one. Capital T small t. From this parent I got capital T. From that parent I got small t. Again the same thing. What do I see over here? All of them are going to be capital T small t. Yeah, all of them are going to be capital T small t. What is this? What does that mean? It means that all of them are going to be tall. How, ma'am? Always understand, whenever you get a dissimilar pair of factors or genes, okay, there will be one that is going to dominate the others. There will be one that is going to be dominating the others. Probably that gene will be able to produce more amount of proteins. That is why I'll say that is more dominating. In that case, when I already know capital T was for tall and small t was for dwarf, and here I already have capital T in all the groups, can I say they are going to be dominant, and all of them are going to be tall? Definitely, ma'am, I can say that all of them are tall. What do you call this as? This is basically the phenotype of the plant. This is the phenotype, the physical appearance. Okay, they look like this. So I say that this is the phenotype of the plant. They are all tall. Now there is something that is known as genotype. Genotype means what? Genetic makeup. This thing that you see over here, right over here, this thing is what? Genetic makeup. They are all capital T small t. By the way, I want to tell you one more very important thing. Whenever you have a combination like this, where both the alleles are same, you say that this condition is known as homozygous condition. Homozygous tall means it will be capital T, capital T. Now in this condition, they are tall. Phenotype is what? Physical appearance. Physical appearance, they are tall. But what is the genetic makeup? Are they similar? One is capital T, another small t. I'll say it is heterozygous. I am going to say it is going to be what? Heterozygous in nature. They are going to be heterozygous. Why? Because you have dissimilar alleles. These are nothing but alleles. One gene, that is height, has two traits, tall and dwarf. That means if I say one gene, it has two things. One is capital T, one small t. These are nothing but they are alleles, subtypes of a gene. Okay, they are going to be subtypes of a gene. Always remember that. Until this particular point, is it clear? I have not completed it. We have to go ahead. Until this particular point, is it clear? Ma'am, KCET second round, when released, tell if you have disturbance. Okay. Iconic second round, they have not published it. I'm assuming it is going to be late. Okay, they have not published it yet. Guys, if it's clear, can you give me a thumbs up, please? Can you give me a thumbs up? I am saying that I have crossed homozygous tall and homozygous dwarf over here. I got heterozygous tall. Okay. Akash Shirade said okay. Fine. Now what I'm going to do is I am going to cross both of them. Okay, I am going to cross both of them, as in basically the progeny is out there, and it goes like this, where it goes. Okay, so I'm going to have to make it over here. I cannot bring back the slide. So right now I have the progeny which looks like this, capital T small t and capital T small t. Are they homozygous or heterozygous? First of all, are they homozygous or heterozygous? Shankar is also there. Hello, hi. Okay, it's okay, Baba, no issues. Homozygous tall or heterozygous tall? Guys, they are the parents, and I am asking you something. Are they homozygous or heterozygous? They are going to be hetero, no? Hetero, why? Both the alleles are different. This allele and this allele is different. Similarly, this and this allele is different. Yes, Raju, they are going to be heterozygous. Okay. Now I am going to form the gametes. This time the gametes are going to be different. Some gametes are going to carry capital T. Some gametes are going to carry small t. Similarly over here, some of them capital T. Some of them are going to carry small t. These are gametes. Write it like this only. Now what? Now we are going to cross them and get the next generation, that is F2 generation. When we cross them, always understand, over here I have a gamete, male gamete. This fuses with the female gamete. I get capital T, capital T. Over here I have the male gamete. This can fuse with the female gamete over here, having small t. Okay, here I have a male gamete, here I have a female gamete. Anyway, we are going to write the dominant character first, so I'll write it like this, capital T and then small t. Then we have this and this, small t, small t. Now I am asking you a question. What is the phenotype? What is the phenotype of this particular plant? Both of them are capital T, capital T. Since both of them are capital T, what is the phenotype going to be like? Physical appearance. How do they look? We do not see genetic nature. No, we cannot see genetics of it. We can only see the physical appearance. Motivational, tall, exactly. This is going to be tall. What about this? 3 tall and 1 dwarf. Everyone says tall. So this is going to be tall. This is going to be tall. This is the one which is going to be dwarf in nature. The phenotypic ratio, if at all I've been asked about, please tell me the ratio of the phenotypes. Phenotypic ratio. Can I say, in my appearance, 3 of them are tall and 1 of them is going to be dwarf? Always write your answer like this, and I'll say recommended way to write your answer is 3 tall is to 1 dwarf. Now if I'm asking you a question, what is the genotypic ratio? 3 is to 1. Bhakti, Mehrun, motivational, perfect, everyone. Pavan, everyone, Raju. Okay. Genotypic ratio is 1 is to 2 is to 1. Is it 1 is to 2 is to 1? Let's check it out. So genotypic ratio, if at all we had to calculate, one of them is capital T, capital T. None of the other genotypes looks like this. Genetic makeup does not look like this. So I'll say 1 and then in bracket I'll put capital T, capital T is equals to 2 of them is capital T, small t. So I'll write 2 and then capital T, small t. 1 of them is small t, so I write 1 and then small t, small t. This is going to be what? Genotypic ratio of your F2 generation in one gene inheritance or monohybrid cross. Why did you call monohybrid? Because you're talking about only one character, that is height, and now we have got the hybrids out there where both tall and dwarf have come together. That is why monohybrid cross is being mentioned over here. Cool. Is it clear? Perfect. Motivational, Bhakti, Raju, Pavan, everyone, Mehrun, you guys are rocking. Okay. Okay, now I have a question to you guys. Okay, so after Mendel did all this experiment for the one gene inheritance, he came up with 2 laws. Can you name the 2 laws? Can you name the 2 laws out there? After the one gene inheritance thing, he came up with 2 laws. And by the way, everyone who have not hit on the subscribe button, make sure you hit on the subscribe button, also on the like button. It increases the visibility, okay? Law of inheritance. It's Mehran ma'am. I'm so sorry. Okay. Laws of inheritance. Yeah, macha. Which law of inheritance? There are two laws of inheritance that is said. Yes, law of segregation, one, and the other one, the first one was what? The first one is going to be what? Apart from law of segregation? Apart from Pavan, I'll try to complete. Okay, I'll try my best to do so. Law of dominance, exactly. So listen to me very, very carefully over here. Listen to me very, very carefully over here. The law of dominance says what? Law of dominance says that in a pair of dissimilar factors, ambaba it went, okay. Law of dominance says that in a pair of dissimilar factors, suppose like this, one is going to be dominating over the other. One is going to be dominating over the other, and the one that is dominant is known as the dominant allele. It is known as the dominant allele. And the other one which is there, it is going to be known as what? The recessive allele. This is known as the law of dominance, which he gave after this. In the first generation, basically he did not see anything. Why? Because of law of dominance. All of them were like this. They were all appearing to be tall. Why? Because of the law of dominance. Okay. In the next generation, he thought that there were no dwarf ones, but in the next generation he was able to see the dwarf ones. Why? Because one thing was happening out there. What was that one thing? Basically, when the alleles, they are there, the factors are there, when they form the gametes, they are going to segregate. They are going to segregate. Every time randomly one is going to go to each gamete. One is going to go to each gamete. This is known as law of segregation. Okay, while the formation of gametes happen, what is going to be there? When a pair of alleles, one allele go to each of the gametes, this is known as law of segregation. If segregation was not happening, Baba, they will all come together only. The chromosomes will be coming together. Okay, it is important for KCET. Yes, dear. Cool. So this is how it goes, and I think this part of the story is super clear. I hope you will be able to go ahead with the schematic representation of one gene inheritance. And here, by the way, this is the Punnett square picture. Here you have tall, here you have dwarf. You have got the gametes capital T, capital T. Here you have got small t, small t. When you cross them, all of them are going to be capital T, small t. That means all of them are going to be tall only. Okay, that is law of dominance that is being shown over here. Now from the tall plan, when you cross, you are going to see that three of them are going to be tall, one of them is going to be dwarf. This is the phenotypic ratio. Here you have proved the segregation as well. So Pavan gave me yes ma'am. Others children, if at all the first part is clear. Schematic representation of one gene inheritance. Can you give me a thumbs up? I'll request you to give me a thumbs up. That becomes more easy for me rather than reading the whole thing. Okay. Now over here, I have a question which is almost similar. Thank you, Raju. Okay, which is almost very much similar to what we have discussed right now, and it applies the same logic over here. Crystal clear. Thank you so much. A pea plant with purple flowers was crossed with plant having white flowers. Listen to me carefully. A pea plant having purple flowers were crossed with a plant having white flowers. The progeny produced only purple flowers. What do you understand from that? If I say purple flowers were crossed with basically white one, and I am saying that we get only purple ones. I am saying we get only purple ones. What do you understand from that? Purple is the dominant one, right? Purple is the dominant one. The first generation, if all of them are purple, it's the dominant one. On selfing, the plants produced 482 plants with purple flowers and 162 plants with white flowers. Explain the mechanism that accounts for these results. That means you're talking about then 482 is a more number and 162 is a lesser number. You're saying 482 are purple, okay, and this one is going to be the white ones. First of all, you got all the purples, but when you cross the purple, you got something like this. What ratio is this? This is basically nothing but 3:1 ratio. How do you explain this answer? Okay, how do you explain this answer? Let's go. Oh my God. Yes. So according to Mendel's law of dominance, the purple flower allele expresses itself in the presence of the white allele. So over here, when I talk about all of these general things out there, I can assume, I can give the term P for the purple and small p for white. I can say probably we started with something like this. When they crossed, all of them were something looking like this. All of them were purple. So according to Mendel's law of dominance, the purple color flower expresses itself in the presence of the white allele. Done. Hence the cross PP plus into PP produced all purple flowers in the F1 plant. Done. Explain the mechanism. First part is done. Now according to Mendel's law of segregation, the next law, the two alleles of a gene separate during gamete formation. They are going to separate during gamete formation. Half of them will carry small p, another half will carry the big P. Either way, similarly here also in the same way happens. If segregation is not happening, you are not going to see them coming together to form white flowers. You will not see that, or you will not see both of them coming together to form the purple flowers. But more majorly the dwarf ones, or not dwarf ones, the recessive character is going to just go off. So always remember, it's very important. According to Mendel's law of segregation, the two alleles of a gene separate during gamete formation. On selfing, the alleles recombine to produce the F2 phenotype ratio of 3 purple and 1 white. The observed ratio 482 to 162 white confirms this Mendelian laws out there. So these laws are being confirmed out there. Will you be able to answer questions like this if we apply a little bit of logic here and there? Right? Clear? No. Will you be able to answer these type of questions? If yes, give me a thumbs up, Baba. If you have any doubt that is there in your head and says that, Ma'am, I cannot do it, so then tell me. Yes, Pavan, law of segregation does not have any, exceptions out there. Perfect. Guys, clear? Thumbs up, thumbs up, thumbs up. Yes, perfect. Okay, now we are coming up with one thing, that is law of dominance we studied. If capital T, small t is there, then basically everything is going to be dependent on the dominant one. Here I am going to talk about an exception. Okay, this is also a very important question. You can be asked like this: schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as an example. What do you mean by incomplete dominance? So I'm going to talk about it right over here. Suppose there is a plant that is having red flowers. Okay, suppose there is a plant that is having red flowers and there is a plant that is having white flowers. White flowers. Okay, we thought that red is dominant one, basically, which is why we mentioned it with capital R. And this one, the white ones are mentioned with small r. We have to show the schematic representation, so we have to go. Okay, sorry, Bhakti. Ma'am, can you explain the law of segregation once again? Yes, law of segregation, guys, wait. Law of segregation basically means that whenever you are going to form the gamete, okay, whenever you are planning to form the gamete, the alleles which are there, these are the alleles, they are going to separate. They cannot come together. They will be separating down, and one allele goes to one gamete. One allele goes to one gamete. Only when that happens, basically this guy also will get an equal opportunity, should show up its character later on. Suppose this guy comes with another small t, it will be able to show its character, right? So law of segregation means when you are forming the gametes, both the alleles, from a pair of alleles, the pair of alleles, each allele is going to segregate into each gamete. In general, basically I mean that when you talk about chromosome, a pair of chromosome, in one gamete only one chromosome goes, in another gamete only another chromosome goes. So in this gamete you had the capital T gene, in here you had the small t gene. So you can say this one has capital T and this one has small t. That is what I mean. Okay, they are going to be divided. Breakup is going to happen over here. Okay, and they cannot go together. If at all they go together, we will get extra chromosomes. What is our major aim? If at all you see, we have 46 number of chromosomes. By that I mean 1, 2, 3, 4, 5, 6, 23 here, and basically 23 here also on both the sides, right? That is how it goes. Now when we form the gamete, only half of it is going to come to a gamete, half of it will go to a gamete. Why? Because we need only 23 chromosomes. If a male has 23 and the female has 23, these 23 will come together and again form this 46 number of chromosome. And each chromosome which I get from the male, similar thing I will have from the female also. They might have the same allele. Here it is capital T, they can also have capital T, but it can be small t also. It can be anything. That's the logic. Okay. Is it clear? What is, okay, yes ma'am, but I always get confused between terms alleles and genes. Okay, I'll tell you one thing. So, I am saying, you look at this chromosome very clearly, okay? This is a chromosome, I am saying there is a gene These two are genes, okay? These two things that you are seeing are genes. Genes for what? Genes for height. Okay, cool. Now suppose here there is capital T, here there is small t gene. That means G's gene codes for lesser protein. This gene codes for greater amount of protein. How do I say that there are different type of genes? I have to call them something. These different type of genes that I have, they are known as alleles. They are known as alleles. So this is one type of gene, allele T, and this is another type of gene, allele capital T. The type of genes which are there, they are known as alleles. That's all. As simple as that. Cannot go way beyond it. Okay, or we can say, suppose we say height is a character. Under the height you have trait tall and dwarf. These are two segregation of the same thing, height. Similarly, of one gene, the different types of it are known as alleles. It can be of the same type, capital T, it can be of the different type, small t, capital T. Okay, is it clear now? And everyone, so for example, I marry someone, okay, I plan to have a baby later on. Fine, my chromosome, only one chromosome will be there for one pair, okay. Suppose this is the first pair of chromosome. From my body, I will be having this chromosome, having a gene here for height. Similarly, from my partner's body, also there will be another chromosome having a gene here for height. These two different type of genes that I am having, they are known as alleles, and you will be having them in the same location for everyone. Cool? Fine. Now let's go. Okay, we were talking about the incomplete dominance part. Listen to me carefully. I have the red flower and I have the white flower. Red plant and the white plant. Red is denoted like this, white is denoted like this. Okay, so these are what? The parents. Now we are going to form the gametes. Gametes will have only half the number of chromosomes, so either it can be R, another also, there is no choice, it will be R only. Similarly, here it will be small r, here also it will be small r. In the F1 generation, the first generation, what do you get? In the first generation, this and this crosses, you get capital R, small r. This and this crosses, you get capital R, small r. This and this crosses, capital R, small r. This and this crosses, capital R, small r. How many capital R, small r? So many capital R, small r. Now I want to ask you a question. What is the genotype over here? First of all, tell me, what is the genotype over here, children? Come on. What is the genetic makeup over here that you can see? Genotype, not the phenotype. I'm asking about the genotype. In the first generation, for all of them, what is the genotype? Come on, come on, come on. Pinka, I'm asking about genotype. Chandrasekhar, 1:3:1. Genotype. I'm talking about genotype. What is a genotype? It is capital R, small r. Genetic makeup. Genotype means genetic makeup. It is capital R, small r. Okay. Now I'm asking about the phenotype, physical appearance. What is the physical appearance? How do they look? What do you expect? Capital R, small r is there. Capital R should be dominant, which is why it is written in capital letter, right? What do you think is the color of these flowers which are going to be formed? Now pink, ma'am. Yes, it is going to be pink in color. Did we cross any pink flowers over here? No. What did we do? We had red flowers, we had white flowers, we crossed them. Then how can you get pink? Baba, because of something that is known as incomplete dominance. You see this because of what? Incomplete dominance. What is exactly happening up over here? This capital R is not be able to completely be dominant over the small r. Basically, it is probably not able to do its whole function, or is not able to produce its entire protein that is required over here, which is why I'll say it is incomplete dominant. That is why you will get a middle color, which is what? Pink. It is going to be pink. This is the first generation. Then the people were like, oh my god, how can this happen? This never happened before. This never happened before. So again a cross was being done. This time the cross was between the siblings itself. So over here what was done? Suppose the parents are capital R, small r. Again capital R, small r. What will be the gametes? Gametes will be capital R. Gametes will be small r. Here gamete will be capital R. Another gamete will be small r. Right, done. Now we are going to fuse them. F2 generation. This and this comes together, I get capital R, capital R. This and this comes together, I get capital R, small r. This and this comes together, I get capital R, small r. And finally, this and this comes together, I get small r, small r. Right? I am asking you a very important question. I am asking you a very important question, that this time when you get something like this, capital R, capital R, capital R, small r, capital R, small r, small r, small r, what do you expect? What is going to be the phenotype like? What do you think is going to be the phenotype like? Any idea? So over here, I can just make it like this. You have capital R, you have capital R, small r. This is your parent. Okay, this is your gamete. Please write it like gamete itself. Capital R, small r. Capital R, small r. And now when I fuse them, F1 generation, this and this fuses, I get capital R, capital R. This and this fuses, capital R, small r. This and this, capital R, small r. This and this, small r, small r. First of all, tell me the phenotype of it. Tell me the physical appearance. Okay, so Pavan says 1 red, 2 pink and 1 white. Okay, so this one which is there, Baba, it is going to be red. Am I assuming correctly? This is going to be red? Yes, this is red, ma'am. What about this one? What about this one? This is also going to be red. That is how generally it works, right? Capital R dominant over R. No, Baba, it is going to be pink in color, incomplete dominance. Similarly, this one is also incomplete dominant, you will get pink color. And finally, what happens to this one? What happens to this one? This is going to be white in color, smaller, white in color, no one to dominate. It is going to be white in color. Bhakti says 3:1, motivational says 1:2:1. The ratio, if at all, I check it out. Phenotypic ratio, it is going to be 1 red is to 2 pink is to 1 white is to 1 white. Now who said 3:1? For 3:1, children, listen to me carefully. Over here you are not going to see 3:1. Phenotypic ratio is 1:2:1. What about the genotypic ratio? Genotypic ratio you check. Genetic makeup ratio. Genotypic ratio, it's going to be even different. Okay, one is going to be capital R, capital R. Does anyone has capital R, capital R? No. What about this one? This one is going to be, 2 of them are going to be capital R, small r, and 1 of them which is there is going to be small r, small r. Again, the ratio is 1:2:1. So always, yes, both genotypic as well as phenotypic ratio is going to be 1:2:1. Motivational, perfect. Bhakti, Baba, did you get it? Did you get to understand why and things like that? But R is dominating. No, R is not dominating. Why? That is what I was talking about. R is going to be showing incomplete dominance. This is an exception that we have. Usually everything follows law of dominance, but you have exceptions in the law of dominance are there, and this is one exception where R is not able to be completely dominant over small r. That is why it is going to be pink in color. Okay, it is going to be pink in color. This is an exception of your first law of inheritance, that is law of dominance. And this is seen in what? Snapdragon plant, which you also call as Antirrhinum majus. Ma'am, Mehreen, can you give important questions for these 3 chapters? Dear, I had made a separate video on this. Can you please check it out? It's there in our YouTube channel. Okay, principles of inheritance and variation is left to be done. Tomorrow we will be getting that particular video also. Okay? 4 o'clock plant, yes. Motivational is also known as 4 o'clock plant. Perfect. So this is how we have to go. I hope this is clear. Make sure you understand that both their ratios are going to be same. This was my favorite chapter, and it is also the chapter I started preparing seriously. That's so nice. Okay, now let's go ahead with the next question. Can we? Guys, can we go ahead with the next question? If yes, can you give me a thumbs up? If it's okay for me to go ahead with the next question, can you give me a thumbs up? Now this is going to get trickier, this part of the question. Okay, we are going to go ahead with, yes, perfect. Thank you so much, motivational, Pavan and all. Define the law of independent assortment and represent schematically the result of two gene interaction by taking seed shape and seed color in pea plant. Now from here, you have to listen to me very, very carefully, otherwise we might not be able to understand anything. So until now, we were talking about what? Monohybrid cross. One character was being taken. Right, one character was being taken, and that is height. It was okay, but now on we are going to go ahead with dihybrid cross. What is the name of this cross? We call this as dihybrid cross. What do you mean by this cross? We are going to take two characters at once. Okay, so over here, how do I understand it's dihybrid cross? Because it is clearly mentioned, represent schematically the result of two genes. Okay, two genes that is responsible for two character interaction by taking seed shape, one character, seed color, another character out there. Now in that, always remember, you have the dominant character round. Okay, round. In terms of seed shape, round is a dominant character. In terms of seed shape, wrinkled is a recessive character. It's going to be recessive character. Okay, in the, as I said, it is about the shape. I'm talking about the shape. This is going to be dominant, this is going to be recessive. Okay, what about the color? In color, yellow is going to be dominant and green is going to be recessive. Okay, yellow, which is there in the terms of color, it is going to be dominant, and over here the green one is going to be recessive. How do I explain that? Listen, for round, what do I say? For tall, what did you do? Capital T, capital T. And when you're seeing pure breed means homozygous. You're talking about homozygous. Okay, they are going to be same. For round, I can mention like this, capital R, capital R. For yellow, again they are homozygous, so I'll mention like this, capital Y, capital Y. Similarly, for wrinkled, I will be making the smaller letter for this, small r, small r. Okay, and for green, I will be making the smaller letter for Y, that is small y, small y. Done? Done, ma'am. Is it clear? This is what? This is the parent. This is basically the parent that I have. Now I want to form the gamete. How do you form the gamete over here? So for that, I am going to explain you something which is going to stay with you throughout your life if at all you listen to me carefully. Okay, it's going to be very, very easy. So listen to me carefully. Over here, just imagine you have a chromosome, another pair of chromosome right over here. This pair contains gene for what? Shape. This pair contains the gene for shape, and both the alleles are same. Here is capital R, here also it's capital R. In this case, okay, this is another chromosome that we have. Here there is a gene for color. This is Y and this one is also Y. Similarly, I'm going to make something over there also. You have a pair of chromosome, okay, where there are two alleles, small r, another pair of chromosome for the color, there is small y and small y. Now when you say that, ma'am, I have to form gametes, how do I form the gametes? Listen to me. From this pair, only one can come. From this pair, only one can come. So suppose let me write capital R. So from this pair, only one can come. Suppose can I write capital Y. Whatever I do over here, I am going to get these type of gametes only. I will be getting 4 of them. I'm not making all 4 of them, because whatever you do, you will be getting that only. From here, whoever comes, it will be capital R. From here, whoever comes, it will be capital Y. So this is the combination that I have. Similarly, from here, whatever comes, it will be small r. From here, whoever comes, it will be small y. So all the gametes are going to be like this only, right? Right, ma'am. Okay, now we want to fuse the gametes. Suppose there are multiple gametes. I've just made one and we are fusing them. When we fuse them, what do you get? Capital R, small r, capital Y, small y. But how do they look? That is more important. How do they look to you? They look like this, Baba. Over here, the chromosome that came from here carrying capital R, the chromosome that came from there carrying small r. This is what they look. Over here, when I say capital R, small r, this is what they look. Similarly, from here I got capital Y, from there I got small y. This is the chromosome that looks over here. So all of them that I have got in the first generation, how are they going to be like? They are going to, Raju says round and yellow. Is it going to be round and yellow? Yes, the capital R is dominating over small r, capital Y is dominating over small y, which is Y. When it dominates, I'll say round. When it dominates, I'll say yellow. All of them are going to be round and yellow. Is it heterozygous or homozygous? The question is it heterozygous heterozygous over here or is it homozygous? Can you tell me that? Are they heterozygous round and yellow or homozygous round and yellow? Any idea? Heterozygous round and yellow, heterozygous, right? Exactly, motivational and Pawan. They're going to be heterozygous. Why? Homozygous looks like this. Both the alleles should be same. Here they are heterozygous in nature. Perfect. Now I'm going to go ahead. Okay, I will be going ahead. I'm going to cross something like this. I'm going to cross them. Okay, I've got the progenies. I'll be crossing them. Let's do that. So over here, I have capital R small r and capital Y small y. Here also I have capital R small r and capital Y small y. Okay, okay. Now use your imagination again. How is the chromosome like? One is capital R, another one is small r. One is capital Y, another one is small y. Same thing happens over here. Here also, one is going to be capital R, another is going to be small r. One is going to be capital Y, another one is going to be small y. Now when I form the gametes, how do they look? How do they look? Let me check it out. When I form the gamete, from this pair only one can come. Law of segregation. If you remember, law of segregation, from a pair of allele only one can come. So I have made this. From this pair of allele only one can come. This is one type. This is just one type. Similarly, suppose again from this pair of allele capital R came, from this pair of allele small y came. This is another type of gamete. This can also happen. Okay. Now suppose from here small r came this time, and from here capital Y came. This is another type of gamete. Now from here small r came, from here came small y. This is another type of gamete. How many type of gametes did you form over here, Baba? You formed 4 gametes. Will you get the 4 gametes here also? You definitely will get the 4 gametes here also, because it's also the same thing. Nothing changed, no changes there. Now what do? When you have got the 4 gametes here, you cannot cross them and do like this. That will be way too difficult. That is why the teacher suggests go ahead with making Punnett square. So I'll make the Punnett square. Here I have got the gametes. Same gametes I'll be getting over here. Capital R small, capital R capital Y, capital R small y, small r capital Y, and small r small y. Right? Now when I cross them, when this crosses with this, both of them capital R, capital Y, capital Y. When this crosses with this, what do I get? Capital R, capital Y, small y. When this crosses with this, capital R, small r, capital Y, capital Y. Finally, when this crosses with this, capital R, small r, capital Y, small y. This is how you have to keep on going. You have to keep on going. By the way, generally no one asks you to make the entire thing like this. If at all they ask you, you can easily make things. And always remember at the end, what do you get? That is even more important. Okay, at the end, what are you going to get? That is one thing that is most important. You are going to get a ratio. First of all, they will not be asking you about the genotypic ratio, because it is obviously there are, so 4 into 4, I'll say 16 progenies are going to be produced. Okay, 16 progenies are going to be produced, and for remembering each of their, genetic makeup is going to be different. You can just cross and see it. The thing that you usually ask is what? Your phenotypic ratio. How do they appear? So for that, understanding that, Raju, 9:3 to 3:1, exactly. So phenotypic ratio goes like this. It is what? 9:3:1, and the easiest way to remember is 9 of them, basically that's the highest number. It's going to be showing the dominant character. What was the dominant character, Baba? The dominant character was round and yellow. One which is there, it is going to show the recessive character. What was the recessive character that we started with? It was wrinkled and green. Now, ma'am, how do I remember both of them? So first of all, before I go ahead, out of 16 of them, if at all you see 9 plus 3 plus 3 plus 1 is equals to 16, right? Out of 16 of them, 9 plus 1, 10 of them are showing parental character. That means they were showing the parental character. Parents were round and yellow, and here parent were wrinkled and green, right? So 10 of them are showing parental character. Always remember. What about these 3 things? These three things that we see over here, they are going to show some new characters that we did not see until now. So until now, round was with yellow, right? This time round is going to be with green. It's going to be with green. Similarly, wrinkled was with green always. This time wrinkled, which is there, it is going to be with yellow. So these 3 plus 3, what? 6 of them are going to be non-parental in nature. Always remember, they are going to be what, Baba? Non-parental. They are not going to look like the parents. They are recombinants out there. What do you call it? Recombinants, new combinations. Okay? Whereas 9 plus 1, which is there, 10 of them, they are going to be parental. They are going to be parental. Always remember, what is this ratio? Phenotypic ratio. Okay. Ma'am, quarterly paper board in the birthday. Yeah. Yes, dear, these type of questions could be coming up there also. Got it? Children, did you get it? So if at all you are asked, out of 16, how many shows parental character? What will you say? 9 plus 1, 10, ma'am. Okay, how many shows non-parental character? Ma'am, I will say basically 6, ma'am. Right? So that is a basic logic. Now here comes a very important law. After Mendel did the tri-hybrid cross experiment, he came up with a new law that is known as independent assortment. What do you mean by that? But after you listen to that, probably you will be killing me. But still listen to me. Okay? Until now, law of segregation means basically when the gametes are formed, the alleles they separate and they come into each gametes, right? Right. Law of independent assortment means a pair of genes, a pair of allele for one particular character, that is suppose shape, okay, is going to assort independently from the pair of alleles of another character, that is probably what? The color. What I mean by that is, over here, when I say that capital R is coming from this pair, capital R is coming. This pair is for what? This pair is for shape, right? Round wrinkled shape. So when they segregate, capital R has come. Does it say that from this pair I will only have capital Y? Or the capital Y says that I will only go with capital R? Does it say like that? No. They are going to assort independently. Once the capital R goes with capital Y, here the next gamete which is there is basically with small y. They are assorting independently, they do not assort together. They are going to assort independently, which is why, Baba, since you are assorting independently, that is why you get these many gametes, these many types of gametes. And since you have these many type of gametes, you get new combinations out there in the form of round and green and wrinkled and yellow. Always remember this. This is law of independent assortment, where a pair of alleles for one character is going to segregate independently from the pair of alleles of another character. Have you got it? Have you got it? If at all they were not, if at all capital R was saying I will only go with capital Y, you will not see any of these changes. Is it clear? Children. Macha. Clear. If you have any doubts that you want to ask me, ask. But it's super important, super important. During the formation of gametes, the allele of different genes assort independently of another. So the inheritance of one trait does not affect the inheritance of another trait. Okay? Pavan, surely dear, bye-bye, please take care. And thank you for giving the company. Ayyo, just a second. Guys, I'm hoping that you have understood since you're not replying. So this is how you have to go about it, and it's very important. And I have a question over here, a similar kind of question, and goes like this. Observe the representation of a dihybrid cross and answer the question that follows. So this is how it looks. Okay, general dihybrid cross which is there. Okay, general dihybrid cross which you probably, just a second. Okay, this is how it looks, the normal thing that is there. The question goes like this. Write the phenotypic ratio. What is the phenotypic ratio of a dihybrid cross? Everyone, repeat. Come on. What is the phenotypic ratio of a dihybrid cross, if at all they are asking you directly? Dihybrid cross, okay? Come on, motivational as well as Bhakti. Okay, Raju says 9:3:3:1. Okay, so over here, what is the phenotype? Write the phenotypic ratio. Someone said physical appearance. Yes, it is going to be 9:3:3:1. Okay, identify the homozygous dominant genotype in the progeny. What do you mean by that? Homozygous dominant. Okay, G.D. Gautam is also there, says 9:3:3:1. Hello, and yeah, good answer. 1:2:2, yeah, Bhakti, that is a genotypic ratio. Genetic, no? Genetic ratio. In physical appearance, if at all we see, let's see. Okay, in physical appearance, this is what? Round and yellow, both alleles. This is also round and yellow. Why? Because this is round and this one is dominant. That is why round and yellow. This is also round and yellow because this is dominant over here and this is yellow. This is also round and yellow. But genetically, all of them are same. So genotypic ratio obviously is going to be more of them. But phenotypic ratio is going to be 9:3:3:1. Okay, Bhakti? Cool. Guys, I'm asking you a question. Identify homozygous dominant genotype in the progeny. How do you answer that? Homozygous dominant means what? Homozygous dominant. Dominant characters are what? Round and yellow. And homozygous means they have to be same. Homozygous means they have to be same. So it is going to be like this. It is going to be like this. Agreed? Now I can give you a question. The next question is for you. Identify the recessive genotype in the progeny. Try that. Identify the recessive genotype in the progeny. What will be your answer for that? Identify the recessive genotype in the progeny. You are asked about the genotype. You have to talk about the genotype. What will be the genotype of the recessive progeny? Small r, small y, small y. Arre wow, perfect. You have got it now. Okay, recessive genotype will be small r, small y, small y. You do not have anything else. Dominant genotype, homozygous dominant genotype is going to be this. Heterozygous dominant genotype can be many of them. Homozygous dominant is definitely going to be looking like this. Okay. Cool. The next question, number of parental type of progeny. How many of them are going to be just like the parents? Out of 16 progenies that are being made, how many of them are just going to be like the parents? G.D. Gautam, you are also correct, dear. And King, ma'am, I am unable to download the NEET UG application slip after enrolling my NEET roll number. Dear, I would have to check it. Even I have not got a confirmation about exactly how it is working. Dear, if I find a solution for it, I'll definitely make a video on it tomorrow, okay? Okay, so motivational says 10 of them. Motivational says 10 of them. Number of parental type of progeny, yes, it is going to be 10 of them. If at all you remember, class 9:3:3:1 is there. So basically 9 plus 1, it becomes 10. Now number of recombinant type of progeny. Recombinant type of progeny, how many of them will be there? Number of recombinant type of progeny. Come on. I hope you guys are getting. If you have doubt, please stop me then and there and say, ma'am, can you explain once more? I will do that. Trust me on that. 6 of them are going to be non-parental, exactly. And what are those 6? If we studied over here, 9 plus 1, 10 of them parental, and 3 plus 3, they are non-parental. You also call them as recombinant. Recombinant means new combinations. You also call them as recombinant. Okay? Everyone has understood this. Do you have any doubts, Macha? Can I go ahead? If yes, give me a thumbs up. If you give me a thumbs up, we'll go ahead with the next question. Yes, ma'am. Okay, perfect. Cool. By the way, in principles of inheritance, is the whole thing coming in your syllabus? Is the whole thing coming in your syllabus, Baba? For everyone, principles of inheritance, whole chapter is coming, or is it until some limit? Yes, ma'am. Okay. In that case, I don't know if I've taken a lot of questions. Let's see. Anyways. Okay. So identify the genetic disorders given below. You have to identify and tell which are the genetic disorders which are being discussed over here. Sex-linked recessive disorder occurring in about 8% of males and only 0.4% of females, leading to defect in the cone of the eye. What do you think this genetic disorder is? Sex-linked recessive disorder. By the way, every one of you know what is sex-linked, what is autosomal, what is this and that. Do you need an explanation on that first? Or do you have an idea, total idea about what exactly is this? If you need an explanation, tell me. Yes, ma'am. Okay, so motivational says the answer for the first question is color blindness. Yes, the answer is color blindness. First of all, they have mentioned sex-linked recessive disorder, and you have only two sex-linked recessive disorder. One is color blindness and the other one is going to be hemophilia. And when you're saying defect in the cone cells, that mean cone of the eye, that means it has to do something with the color blindness itself. So the answer for this question is going to be color blindness. It is going to be color blindness. Okay, and why does it affect 8% of males and only 0.4% of females? As I said, it is going to be a sex-linked recessive disorder. So yes, you need explanation. So over here, listen, I am going to provide a little bit of explanation, okay? I think it is definitely going to help in some ways for you guys. So in general, we have two types of disorders, autosomal and, we have two types of disorders. We say it's Mendelian, and the other one which is there, it is chromosomal. Okay, we have Mendelian disorders and we have chromosomal disorders. In the case of Mendelian disorders, we say that arises due to, for example, I have a chromosome over here. Just a second. I have a chromosome over here and there is some issue with the genes on these chromosomes. There is some issue with the genes on this particular chromosome that leads to the disease. That is going to lead to a disease. This is going to be known as Mendelian disorder. On the other hand, when I talk about this one, in the chromosomal case, what is going to happen? Either, okay, suppose I should be having two chromosomes, but I have an extra chromosome. That is going to cause a trouble. Or suppose I should be having two chromosomes, but I just have one chromosome. That is also going to lead to a disease. That is basically chromosomal disorders. So Mendelian disorders and chromosomal disorders are different. Please do understand that. And when I say sex-linked disorder, when I say autosomal disorder, I'm talking about this. I'm talking about this. When I say sex-linked disorders, that means I'm talking about the sex chromosome. In the sex chromosome, the genes that they carry, they have some defects in them. Okay, and when I say autosomal, I mean that in our body, for example, we have 23 pair of chromosome. We have 23 pair of chromosome. Out of that, 22 of them are going to be autosomes. Autosomes basically are the chromosomes which is responsible for all the other characters of our body apart from deciding the sex of us. Okay, the last pair that is the 23rd pair that we have is a pair of sex chromosome. So if at all, if there is some defect in the autosome, in the genes on the autosome, I'll say it's an autosomal disorders. If at all there is some issue with the sex chromosomes are there, in the genes of sex chromosome, I'll say it is sex-linked disorder. Okay, in the genes, I'm not talking about the numbers. Number thing comes over here in chromosomal. Mendelian means something totally different out there. It's totally about the genes. Now, I'm talking about the Mendelian disorders and there are few of them which you need to know. Okay, in Mendelian, you have two sex-linked recessive disorders. Sex-linked recessive disorders. Where what is going to happen is that, suppose over here I'm talking about what are diseases that you have. You have color blindness, that is the first thing that you have, and the second thing that you have is going to be hemophilia. So I'm talking about color blindness first. In color blindness, over here suppose I have a female XX, okay, and there is a male XY. So usually if it's a recessive disorder, that means it will only show the disease when both the chromosomes are same, equally affected. For example, if I say this is a female and if I say her one chromosome's gene is affected, okay, she has a color blindness gene over here, one of her chromosome. Do you think she will be colorblind? Do you think she will be colorblind? Yes or no? I'm asking you, if condition is like this, only one chromosome is affected and it's a recessive disease. Recessive disease only happens when two of the chromosomes have the same condition there. Do you think she will be having the disease out there? She will be colorblind out there? Yes or no? Yes or no? Guys, asking you a question. She will be colorblind? She would not be colorblind. No, ma'am. No. Why is she not colorblind? Because she has a normal chromosome which is going to overshadow the effect of the other chromosome and she will be normal. Is she, what will I call? She is what? She is going to be a carrier. She is definitely going to be a carrier, but she does not have the disease. She can see all the colors very, very nicely. Okay. Now, on the other hand, if I talk about this guy, This guy only has one X chromosome. Okay, this guy only has one X chromosome. If at all I say that this guy's X chromosome is affected, one X chromosome, Papa guy has only one, and that X chromosome is affected, do you think he will be colorblind? Yes or no? This guy who is having X and Y and one of his X chromosome is affected, is he going to be colorblind or not? What do you think? Yes, ma'am. Why yes? The reason being that there is no one to dominate over the other one. If you had a normal X chromosome, it worked. But here that was not the case. That is not at all the case. You get it? That is the reason you have to. So I am going to go ahead and I am going to explain you with different scenarios. I think that is going to have a better understanding. Okay? So this is one thing. I'm just going to remove it and I'm going to show you scenarios. The first scenario goes like this. The female over here is perfectly normal. Okay? Or suppose I'll say she is a carrier and this guy is normal. This guy is completely normal. Suppose this chromosome and this chromosome comes together when they make progeny. What is going to happen? X and X comes together and both the X chromosomes are normal. So this is going to be a normal female. X and X means it is going to be a female. Normal female. Okay. Suppose this chromosome and this chromosome come together. X and Y. X and Y is there. No effect is there. So I'll say it's going to be a normal male. Normal male. Okay, ma'am. Suppose this and this comes together. X and XC. What happens here? This is going to be a normal female. I'll say yes, she's a normal female. Normal, but a carrier. But a carrier, she does not have the disease. She does not have the disease. What about when this and this comes together? Why she does not have the disease? Because there's a normal chromosome to overshadow the effect. But when this and this comes together, XC and Y comes together, I'll say that this particular guy which is there, he has the disease. I'll say he has the disease. He is going to be colorblind in nature. Why do I say that? Because there is no other X chromosome to overshadow the effect out there. So in this case, in the diseases which are sex-linked recessive, if at all it was dominant, then only one chromosome is enough for making the person diseased. Okay, it is one chromosome is more than enough for making this particular person diseased if it was a dominant disorder. But that is not the case. It's a recessive disorder. It will only happen if at all the condition is recessive. Okay, it will only happen to her if at all the condition is recessive. I'll show you that scenario also. So in this case, there is no one to overshadow the effect, which is why the guy is colorblind. Okay, so it is not fair for the guy. Okay, rest of females are quite safe over here. Now the next scenario. Suppose you think over here, there is a female who is again a carrier. She does not have the disease. There is a male who have the disease. There is a guy who have the disease. Let's see the flow. This and this comes together. Is she normal or not? Guys, is she going to be normal or colorblind? Is she going to be normal or colorblind? Yes. Come on. What is the status going to be like? Normal or colorblind? Normal. Why normal, Baba? Because the other chromosome is there. That is why she is not colorblind. I'll say she's a normal female, but a carrier. But definitely a carrier. Okay, but I'll consider this to be normal itself. Suppose this and this comes together. This is going to be normal guy. I do not even have to ask this. This is going to be a normal male. Cool. Now when this and this comes together, XC, XC, she is going to be normal or she is going to have color blindness. This condition, for the first time it happened, is she going to be normal or she going to be colorblind? What do you think? Gautam, motivational. Affected, exactly. She is going to be a colorblind female. Why? Because this is a recessive condition. Both the chromosomes are same. Both the chromosomes are same. they are going to recessive condition, they will be showing the effect. There is no one to overshadow them. Again, when this and this comes together, you get XCY. This guy is again going to be colorblind. So if you see, in general, whatever you do, males have high chances. Why? Because only one affected chromosome has to come over here to make the males affected. That is why we say sex-linked recessive disorders, in that the males are the one who are going to be majorly affected and the females are going to be less affected. So in color blindness, we say 8% chances there for the male to be affected and 0.4% chances there for the female to be affected. When is the female going to be affected? When the condition is like this, when mother is a carrier and the father is going to be colorblind person. When mother is going to be carrier and the father has to be colorblind, then only you will be getting this particular chromosome, right? So please understand that in this condition, or the mother can be colorblind, father can be colorblind, then the progeny will also be colorblind. That is another thing, but in general I was saying. Did you understand the logic? Understood the logic behind it? Now there is hemophilia also. Okay, there is hemophilia also, but in hemophilia we generally say the females are not affected. Okay, we generally say the females are not going to be affected. I'll tell you why. But is this understood? If yes, can you give me a thumbs up? If this is cleared, done and dusted, a thumbs up. If you have doubt, ask me straightforward. Yes, ma'am. Okay, cool. Now we go with hemophilia. Okay, what is going to happen with hemophilia? Let's check that. So in hemophilia, I say this is given in your NCERT. Hemophilia is what? Hemophilia is a disease where this is color blindness. People can definitely live with color blindness. They are not going to die. Okay, it's just going to be a little different for them in terms of color and things like that. Definitely it's difficult, but definitely it's not fatal for them. But hemophilia is not like that. Why? First of all, it is going to be a blood disorder. Blood disorder as in, basically, you need your blood to clot. If at all there, suppose I get a wound over here and I get a cut, there is a blood flow that is happening. It has to stop sometime. If it does not stop sometime, basically my blood goes off, right? So in this disease, that is hemophilia, what happens is that there a clotting enzyme which is responsible for the clotting, okay, for the clotting of blood is being affected. The gene that is responsible for formation of this particular enzyme, a protein, is being affected, which is why there is no clotting that is going to be seen in the people who have hemophilia. Cool? In the people who have hemophilia. Now in this case, when I say, I have a dialogue, generally I have a dialogue which says that females are usually not hemophilic. They can be carriers, but they are generally not hemophilic. What is the reason for that? So there we saw in color blindness the ratio was less. Here I am saying that there are very less chances. I'm not saying there is no chance. I am saying there are very less chances. Hello Hanamanta. Okay, I'm saying there is very less chances for the female to be affected. The reason is that, listen to me very carefully. Over here, XX, okay, XY. Cool? In this particular case, if it's like this, this and this comes together, I get XX. She is going to be normal. This and this comes together, I get XY. He is also normal. This and this comes together, again the same thing. She is going to be normal. She is just going to be a carrier. Why? Because she has a normal gene. Cool? That is clear. Now when this and this comes together, what is going to happen? He is going to be affected. He definitely is going to be affected. Why? Because he does not have any other chromosome to overshadow the effect. Now the problem is, this guy who is hemophilic, cannot stay until, or probably might not live until the time he might be able to reproduce. Okay, people before, I do not know, like as of now there are so many medicines and things like that, but before it was not like that, which is exactly why people, or generally men who has hemophilia, will not be able to live until the reproducible age or reproductible age are there. In that particular case, if he is not able to stay until that particular age, how will he produce a female who is having the disease? Because if at all a female needs to have the disease, it should be like this. One X chromosome should be coming from the mother, but the other chromosome should be coming from the father, right? It should be ideally coming from the father, but this case is very, very less. The chances for that to happen is going to be very, very less. So do you think it is possible? What do you think? Do you think it is possible for a female to have the disease? The answer is very less. The answer is going to be very less. So hemophilia is going to be a different disease, as I said, apart from color blindness. It is going to be fatal, which is why we expect that the male will not be much, male will be affected, but the females generally are not going to be hemophilic. Got it? Understood the logic? Or doubt is there? If you have understood, do let me know. If you have not, do let me know about that also. Then we will go ahead to the next question which is there. Understood. Okay. So that was about the sex-linked diseases which were there. Okay, 2 diseases are there, that is hemophilia as well as color blindness. Now we're going to talk about the autosomal diseases. Okay, that means the genes on the autosomes are being affected. You have 3 diseases in your NCERT. One is going to be phenylketonuria, other one which is there is going to be sickle cell anemia, and the final one which is there is going to be thalassemia. These 3 are autosomal disorders. That means normal chromosomes, normal autosomes, their gene are being affected. In phenylketonuria, there is a gene. Okay, first of all, what happens in general is that you have phenylalanine in your body. You have phenylalanine, which is actually an amino acid which is there in our body. This phenylalanine which is there, it has to be converted to something that is known as tyrosine. And for this, you need an enzyme. You need an enzyme whose name is sorry. Oh my God. Oh my God, what is the name of the enzyme? Phenylalanine. Okay, the name of the enzyme is phenylalanine hydroxylase. Now this particular enzyme is being coded by a gene on what? On the autosome, but somehow there is some effect over here, there is some mutation over here, which is why this particular enzyme is not being produced. Now when this enzyme is not being produced, that is phenylalanine hydroxylase is not being produced, what happens, Baba? There is going to be a problem. Okay, there is going to be a problem, and that problem is the phenylalanine does not get converted to tyrosine, rather it accumulates in the body in the form of phenylpyruvic acid. It stays in the body in the form of phenylpyruvic acid. This phenylpyruvic acid accumulates in the brain and can cause mental retardation. It can cause mental retardation. Okay, apart from it, there is one more very, very important thing that is happening over here. This tyrosine which is there is super important for our skin pigmentation, or basically this tyrosine helps in the formation of melanin, which is super important for our skin pigmentation. Now when it is not there, what is going to happen? There will be reduced pigmentation. Reduced pigmentation. Your hair as well as your skin will be having reduced color. You might be seeing the plain batch colors out there. That is because of what? Phenylketonuria. So again, as I am saying, this is what? Autosomal recessive disorder. Okay, it is autosomal recessive disorder. Only if 2 recessive genes comes together, that's when this effect is going to be seen. And over here, phenylalanine hydroxylase is not being produced, which is why the conversion of phenylalanine to tyrosine does not take place, which is why it gets accumulated in the body in the form of what? Phenylpyruvic acid, and this phenylpyruvic acid can lead to what? Mental retardation. This accumulation of phenylpyruvic acid can lead to mental retardation. Second thing, tyrosine. Tyrosine, if at all it is not there, basically you need melatonin, and for the formation of that you need tyrosine. If it's not there, it leads to reduced pigmentation of both your hair as well as skin. Okay, am I clear until this particular point? Excreted through urine. Yes, exactly. Now this particular phenylpyruvic acid which is there, it has very less absorption power. Basically it is not getting absorbed in our body, so it is usually released through urine, which is why our urine will also have a very foul smell. Okay, which is why the urine will be having a very foul smell, excreted through urine. That is also there. Clear about it? Any doubt that you are having until this particular point? Any doubt? I also want to talk one more thing about this phenylketonuria, then it will be done. Clear, 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 clear? Children, clear? If yes, please give me a thumbs up. If no, please ask me the question. Yes, ma'am. Okay. Now listen to me very carefully. Okay. This is also an example of something that you know that is known as pleiotropy. You know what's pleiotropy? So usually we have seen that one gene, for example, when I say capital T gene, that is responsible for height. But here, there are certain genes which is responsible for multiple character, not just one character. Okay, so in capital C is there, it is there for height. But over here, when I talk about this particular gene, that is phenylalanine hydroxylase, the gene coding for this, it is responsible for multiple character, not one. For example, it is responsible for conversion of this to this, right? And also, it is responsible for pigmentation. It is also responsible for pigmentation, that is tyrosine. If it was normally produced, basically it is going to help with pigmentation. So one gene has having multiple effects, which is why this is an example of pleiotropy. This mechanism where one gene has multiple effects, please remember, this is known as pleiotropy. And the example for this is phenylketonuria. Example for this is basically phenylketonuria. Okay, got it? Since this gene has multiple effects, that is why multiple things are being affected when someone has phenylketonuria. Yep. Okay, now coming ahead with the next, now going ahead with the next part, that is known as sickle cell anemia. Again, this is going to be an autosomal recessive disorder. It's going to be autosomal recessive. Here also autosomes are going to be affected. Now there's one very important problem that is seen over here, that is, you know, hemoglobin. Hemoglobin, which is responsible for transfer of oxygen and things like that, it is made up of two chains, alpha chain as well as beta chain. Alpha chain as well as beta chain, right? Now I am saying the chromosomes which is responsible for, the chromosome which is responsible for formation of this beta gene, suppose they have some issues, there is some mutation happening over here. Do you think that this beta gene is going to be formed normally? Do you think in that case this beta gene is going to be formed normally? The answer is no. The beta globin chain is going to be formed abnormally, or it's going to have a change structure. Why does that happen? In general, because of a single mutation that is taking place, and this is very, very important. When I say a gene, I mean that basically the gene has multiple codons, okay? They code for certain things. So similar codon that we have over here is GAG. And GAG codon codes for a amino acid. All the things, okay. Motivational, no, is as in you do not have any doubt, is what I am assuming, okay? Right? So anyways, I'm coming back over here. So when I say this gene is mutated, I mean something like this. Every gene which is there, it is made up of multiple codons, and each codon which is there, Baba, it codes for amino acid. It codes for amino acids. So similarly, you going to get so many multiple amino acids, and all these multiple amino acids together come to form the protein. That protein in this particular case is what? The beta globin. Here I am saying that usually it is GAG. The codon that we have over here is going to be GAG, and it's coding for an amino acid which is known as glutamic acid, okay? But because of some mutation, what happened? Instead of having GAG, now you have GUG. Now you have GUG, and this particular codon is something different, is going to code for something else which is known as valine. What are they? They are amino acids only. So instead of having glutamic acid over here, you would now have valine. So beta globin chain is produced or not? The answer is yes, it is being produced. But is it a normal one that we have over here? No, it's not a normal one. The shape of it is going to be different. So Baba, the shape, not the shape, the structure entirely is going to be different. Why? Because of one change in the amino acid. One amino acid change and the whole thing changed up. Now the problem is a red blood cell which is carrying this particular hemoglobin, which is made up of this beta globin as well as the normal alpha globin. What is going to be there happening over here? This would be having reduced potential. It is going to change its cell shape into sickle. Why? Because of this particular thing. That is why you call this as sickle cell anemia. The red blood cells shape changes to what? Sickle. And it is basically a qualitative problem. The quality of the beta globin chain is hampered, not the quantity. Beta globin chain is still being produced. Yes, half moon shaped. Exactly motivational. Okay, so please to remember it. And also, if at all you're facing difficulty in remembering GAG, GUG and things like that, you can remember in this manner. G-A-G, G-U-G. In the alphabetic order, which comes first, A or U? It is going to be A. It is going to be A. So initially we have A. Okay. And this is going to be coding for what? Glutamic acid. Finally, this is coding for what? Valine. Again, G comes first and then comes V. So you can always remember GAG was coding for glutamic acid. GUG was coding for valine. And this is actually the 6th codon. Always remember, there are multiple codons I told you. This is the 6th codon, which is this. Okay, where the mutation and everything is happening. Now when it is sickle in shape, always understand, if it is round in shape, discoid in shape, basically the cells can normally move around. If it is sickle in shape, it can create blockages around your body. And not just that, their oxygen carrying capacity is going to be reduced. Okay, their oxygen carrying capacity is also going to be reduced, which is why it is going to be fatal. It is going to be definitely fatal. Okay, so people can be carriers of sickle cell anemia. They definitely can be carriers of sickle cell anemia. For example, I say that one person has HbA. This is a normal one. Another gene that we have is HbS. This is one gene. This is one allele. This is another allele. Okay, over here. So where do I write it? Okay, so over here again. Just give me a second. Okay, I would have to omit out this part. Then only I can write it. Otherwise everything will go. So over here I can say this is HbA and this is going to be HbS. This is one pair of allele. Again there is HbA and there is HbS. This is another pair of alleles. What is going to happen when this and this comes? I'll say HbA, HbA. It's totally normal. It's totally normal. When suppose this and this comes, first of all, what do you mean by having one S allele? It means that they are carriers. They are carriers, they do not have the disease. Now it can be, Allah bocha. Okay, so basically, just a second, I'm so sorry, yaar. I'm so sorry. In us, normally in us, we can be either like this, or probably we also can be either like this. Will you ever know if at all you're like this? No, you are not going to know. Why? Because you will be a carrier. You have a normal allele that is going to take care of everything. It's a recessive disorder. For you to have the disease, if at all, you need to have the disease. For that, it should be like this. Then only you will be having the disease. So in that case, I am telling you, we can be normal like this. We can be a carrier like this. And for the disease to happen, it should be recessive. Both of the alleles has to be same itself. And how can that be possible? When two carriers marry, that can be possible. So I'll tell you a story. So, you know, in Attapadi region that is there in Kerala and Tamil Nadu and all, you saw one very important thing, or in general also, it was being seen that the cousins, when they marry, their kids have the same kind of disease, or probably they are prone to having diseases. Why is that so? The reason is that probably the cousins who are there, or probably the siblings that is there, they would be having the carrier alleles. They have the same thing. They have the carrier allele. Now what happens? Now what happens over here? When they marry, they are very normal. They do not have the disease. But when they marry, this and this comes together. Okay, there is chance for the baby to be very, very normal. Cool. This and this comes together. Again, there's a chance for the baby to be very normal. Why? Because this is just a carrier. Baby is going to be a carrier only. Now when this and this comes together, HbS comes together, don't you think that the person is going to have the disease? Yes, Baba. When normally this and this comes together, again, when I cross this and this, I get HbA, HbS. So I can say out of 4 children, there is always a chance, 25% chance. Out of 4, I can say 25% chance is there for someone to be diseased if at all both their parents are going to be carrier. And this is one very, very important thing, which is why it is said not to marry between relatives. The reason for this is the thing. I hope it makes sense. Yes? Did you understand? Do you have queries around it? If understood, can you give me a thumbs up? If you have queries around it, can you let me know? Okay, yes, ma'am. Perfect. Cool. Now, last thing that we have in recessive disorders, autosomal recessive disorders, is thalassemia. That is also blood disorder itself. But here we saw it is qualitative, right? Quality was getting hampered. The next thing that we are going to see is quantity is going to get hampered. Over here, you have alpha globin chain. So listen, alpha, just a second. Okay. Alpha is there and beta is there. For that, you have chromosome. The beta globin chain, the beta gene which is there is only one, is at this particular position, HBB. But over here for alpha, you basically have two of them. One is HbA1 and this is going to be HbA2. You have two alleles or you have two alleles over here. Right now I am saying while you make, okay, while you're going to make the chromosomes up over here, okay, or probably alpha and the beta globin chain, suppose this particular gene is being affected. What is going to happen? Still alpha globin chain is going to be produced. Still it is going to be produced. Why? Because you have three of them still left behind. They will manage the work. What if two of them gets affected? Still it will be produced because you have rest two of them. What if this is getting affected? Again it will be managed because one is still left. It can still produce. What if all of them gets going? Basically alpha chain is not going to be produced at all. Similarly in the case of beta globin chain, if one is getting affected, yes, other one is there, it can manage it. Okay, it can manage it. What if both of them goes? There is no beta globin chain that is going to be produced. So that is why I am saying it is going to be a quantitative disorder. Okay, genes can be affected, and basically if lesser number of genes are affected and you have other genes to take care of it, it can be managed. But if all your genes are being affected, basically it cannot be managed. It cannot be managed and the person would be dying off very shortly. That is why people who have thalassemia, who actually have the major form of thalassemia, they will not be living post 10 years of age. It's a quantitative disorder. The quantity of the genes are the problem over here. If little amount of alpha beta chain and alpha is there, that's okay. But if there is no, then that's a problem. Mohantesh, my ma'am, in my that is what I was checking. A lot of people actually in their schools, this particular chapter is not being completed. But anyways, okay, that is why. So don't worry, dear. Lot of them are there where the chapter is not being completed. Until what point has been done? Is the chromosomal disorder done? Oh, sorry, not the chromosomal. Mendelian disorders, all of us completed or not? Okay. Yes, Mendelian is completed, then we can try. Okay guys, now I think we can try to find the answer for all these questions. Sex-linked recessive disorder, we know that it is affecting the cone of the eye. It has to be color blindness, right? We spoke about it. Let's go with the next question. Sex-linked recessive disorder affecting a single protein involved in the clotting of blood. Involved in the clotting of blood. Exactly, it is going to be hemophilia. Hemophilia is a blood disorder and it affects the clotting of the blood. Now, inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. Actually, yeah, accumulation of phenylalanine. Where do you see? It's an inborn error of metabolism. There is nothing. Metabolism means something forming something else, something breaking into something else. So there's an error in the whole process, which is why this particular disease is happening. And not just that, you will see accumulation of phenylalanine. What is this disease? Come on. Guys, what is this disease where you find accumulation of phenylalanine? Phenylketonuria. Yes, you also call this as PKU. This is known as phenylketonuria. Finally, autosomal ringed recessive blood disease where substitution of amino acid in the beta globin chain of hemoglobin is happening. Substitution is happening. So GAG was there becomes GUG, that means glutamic acid gets substituted to what? Valine. What is the disease where you have this substitution being seen? What is the disease where you have this substitution being seen? What do you think is the answer for this question? Sickle cell anemia. Perfect. Oh my God, I feel really happy. Okay, next thing. Autosome linked recessive blood disease, reduced rate of synthesis of globin chain. That means you're talking about quantity of it. Reduced rate of synthesis of globin chain. What do you think is the answer for this? And guys, everyone who is watching this video, make sure you try it out, Baba. Last one. Come on. Autosome-linked recessive blood disease, reduced rate of synthesis of globin chain of hemoglobin. Thalassemia. Yes, it is going to be thalassemia. Perfect. And over here goes your answer. Now I believe that was helpful, which is why the answers and everything came forward. Perfect. Okay, now let's go ahead with the next question. Few autosome-linked recessive blood gene diseases occur in human population. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Explain. So which is the qualitative problem? Qualitative is going to be sickle cell anemia. Quantitative, Baba, is going to be what? Your thalassemia. So you have to give your answer like that. You have to talk about it. Okay, some blood diseases, blah 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 blah. So first one is sickle cell anemia. It's a qualitative problem. Mutation in the HBB gene on chromosome number 11. Oh, by the way, we didn't talk about chromosome number 11. So remember, HBB, okay, beta globin gene is present on chromosome number 11. Okay, and the alpha-1 is present on the 16th chromosome. Beta is on 11th and alpha is on 16th. Cool. So both of them have something like this. It looks like 6, you can remember like that. Almost. Okay, not exactly, but still. Cool. So mutation in the HBB gene causes the production of abnormal hemoglobin. Okay, we got about it. Then this defective hemoglobin leads to sickle-shaped red blood cells, impairing the oxygen transport and causing the blockages in the blood vessels. Here the defect is caused because of the error in the quality, not the quantity, quality of the protein that is being made. Thalassemia is a quantitative problem caused by mutations in the gene responsible for hemoglobin synthesis. It can be HbA1 or HbA2 for alpha chain, or basically HBB for beta chains. There can be mutation in one allele of a gene. As I said, over here it looks like this. Okay, there can be mutation in one allele, that is this one. There are total of 4 of them. It can be 1 or basically 2 or it can be more. So there can be mutation in one allele of a gene or both the alleles of a gene, which leads to reduced or no production of specific hemoglobin. There will be basically either less amount of hemoglobin or no hemoglobin that is going to be produced, leading to what? If hemoglobin is not there, basically oxygen transport, nothing is going to happen. You will feel fatigued. Oh my God, I'm gonna die. You feel like that. Here the defect arises due to errors in the quantity of the gene which is reduced. And by this statement, that is, there can be mutation in one allele of a gene or basically 2 alleles, both the alleles of a gene. I mean this. Okay, so suppose over here this is one gene, HbA1, and this is HbA2. So either one allele or both the allele of a gene can be affected. That is what it means. Cool. This question is clear. Do I need to explain anything here? If no, can you give me a thumbs up? If we can move forward, can we give me a thumbs up? Even though it took a lot of time up there explaining the previous part, but I think that is definitely going to help us in going forward faster. Okay, perfect. Then let's go ahead with the next question that is there. Okay, Mahantesh, give me a thumbs up. Define aneuploidy. Give 2 examples of aneuploidy. Mention the karyotype of Klinefelter syndrome of an affected individual and write their symptoms out there. Write their symptoms out there. So basically, if at all we check, aneuploidy means what, Baba? Aneuploidy basically means either addition or loss of a chromosome. We are basically talking about chromosomal disorders. We are basically talking about chromosomal disorders in that particular case. Okay, and what is exactly happening over here? Either, so you should be apparently having 1 pair of chromosome, but you can sometimes have an extra chromosome over here. Or suppose you should be having 1 pair of chromosome, but you just have 1 chromosome over here. Either of these things can happen. Either of these things can happen, right? This is basically known as aneuploidy. Fine, and 2 examples of aneuploidy. So the cases where basically you get an extra chromosome, this is known as trisomy. And the cases where you're going to lose your chromosome out there, this is known as monosomy. And if you want examples, it's very, very simple. Listen to me very carefully. Okay, there's a disease that we are very much aware of. It is known as Down syndrome. If if you have heard about that disease. What exactly happens over here? I told you we have 23 pair of chromosome, right? So I'm talking right now about the 21st pair of chromosome. This is a pair of chromosomes that we have, and this is what? The 21st pair, right? I am telling, from one parent you should be ideally getting one chromosome. From another parent also you should be getting one chromosome. That's how you get the one total pair. But this is something different. You get two chromosomes from one parent and one chromosome from one parent. That means you have an additional chromosome out there, isn't it? You have an additional chromosome out there, and that is going to cause the whole defect. Ma'am, your voice is not clear. Is it fine now? Is it fine now? Or is it still persisting? Hello, hello. Yes, I believe it's fine now, right? Can I go ahead? Yes, perfect. Okay, so I'm saying that you had one pair, you should be ideally having 21st pair of chromosomes. That means one chromosome from each of your parents. But somehow, because of some issues with the segregation that was happening, okay, probably two chromosomes came into one gamete. So one chromosome came with one gamete. One gamete came with two chromosomes out there, which is why what happened? Now you have three chromosomes for one particular thing. This is causing the disease which is there, which is known as Down syndrome. And I would say it is a trisomy of 21st chromosome. This is what? This is basically caused by trisomy of 21st chromosome. Extra chromosome is out there, and in this particular condition you would be having major symptoms such as mental retardation would be there. Okay, there will be broad palm trees and it will be very significant. There will be congenital heart issues and so many things like that. Okay, furrowed tongue. The tongue is definitely going to be something like this. The tongue is going to be a little bit furrowed, not too much, but yes, there will be furrowed tongue like this and all. Cool. This is basically Down syndrome, and if at all you have been asked about the karyotype of it, how do we explain the karyotype? So Down syndrome, I'm writing what is happening over here. We are basically getting an extra chromosome which is there. This is what? The 21st chromosome. So I'll say it's a trisomy of 21st chromosome. Now how will we write the karyotype of it? So karyotype in general, it goes like this. Okay, so we have almost 46 chromosomes. Not almost, we have actually 46 chromosomes. 44 of them are going to be autosomes, right? 45th chromosome and 46th chromosome which is there, it is going to be the sex chromosome. It is going to be the sex chromosome. So either it can be like this, 44 autosomes plus XY in the case of males, or XX in the case of females. Here in this particular case, I am saying 21st chromosome is extra. 21st chromosome is an autosome or a sex chromosome? Tell me, 21st chromosome which is there, I am saying 23 pair of chromosome. 46 chromosome means 23 pair are there. Okay, suppose this is a 23rd one. 23rd one can be either like this or it can be XY. When I'm talking about the 21st chromosome, is it an autosome or is it a sex chromosome? 21st pair is an autosome or a sex chromosome? Guys, it is going to be an autosome. It is definitely going to be an autosome, right? 21st, 1, 2, 3, 4, 5, 6, that is how we have to count, and that's how we get 46. I am saying the 45th and 46th chromosome, or basically the 23rd pair of chromosome, is basically sex chromosome. Rest all of them are autosomes only. Yes, yes. So in this case, I'll say the number of autosomes is going to increase by 1. I have an extra addition of autosomes. So this is how the karyotype looks like. 45A plus XX, if at all it's a female who is having the Down syndrome, or 45A plus XY if it's a male who is having the Down syndrome. This is the karyotype. This is going to be what, Baba? The karyotype. This is about Down syndrome. Similarly, you have the next thing where you have an additional chromosome, and that is known as Klinefelter syndrome. Okay, it is known as Klinefelter's syndrome. What happens over here in Klinefelter syndrome? This is a sex chromosome related thing. This is not autosome, this is going to be sex chromosome. Over here, it should have been a male. A male means X and Y chromosome should have been there. But now, you are getting one extra chromosome. So one gamete came with two chromosomes. Instead of having one, it came with two chromosomes. You have an extra chromosome over here. This is a sex chromosome which is extra. So can I say it's a trisomy? Again, it's a trisomy, but this time it is going to be of the sex chromosomes out there. How will you say is the karyotype for this? What will be the karyotype for this? Any guesses? Can you try it out? How will you think, or what do you think is the karyotype for this particular thing going to be like? Any idea? Karyotype for this? 44 autosomes remains the same. Autosomes will not have any change. What about the chromosomes, sex chromosomes are there? There is an extra one, and that is the X1. That is going to be the X1. Okay, it will be having extra X chromosome. So in that case, it is going to be 44A XXY. 44A XXY, whatever X chromosome is there extra, you talk about this. Or sometimes, yes, it can be written like this also, 47, XXY. Sometimes they write it like this also. Okay, so that means that 44 autosomes, 45th, 46th, 47th chromosomes are there, and in that the extra one is going to be the X1. That is what you mean by that. Motivational, good one. Okay, now we have one more thing. We would be having the last syndrome that is there, the Turner syndrome. The Turner syndrome. Turner syndrome is basically going to affect the females. What is going to happen? The females generally would be having two X chromosomes, right, Baba? They generally would be having two X chromosomes. So over here, there is one X chromosome. Sorry. Over here, there is one X chromosome. Another one will also be X chromosome. But apparently, there is going to be a loss of a chromosome. There is going to be a loss of a chromosome. A gamete comes with no chromosome at all. In that case, I'll say, is it monosomy or trisomy? Come on, try. Is it monosomy or do you call it as trisomy? Will it be monosomy or will it be trisomy? Come on, come on, come on. Monosomy, exactly. Why? Because you're losing one chromosome out there. And this monosomy of this X chromosome. Again, what will be the karyotype? 44A plus this time it will be XO condition. The condition is going to be what? XO. You do not have anything out there. You get it? Sometimes it is also written as 45. Your total chromosomes which are there are what? This one and then this one. So 45. XO. That is also sometimes written. Either way, please remember this. Monosomy. Akash is coming in break samacha. So this is what it is, and yeah. Now mention the karyotype of Klinefelter's, you know that syndrome of an active angel, and write its symptoms. What are the symptoms? So this guy will be having a masculine body itself, but along with that there will be gynecomastia. There is masculine body for sure, but there is gynecomastia as in formation of breast. Why? Because you have an extra X chromosome. That is why there will be formation of breast. And always remember, in Turner syndrome or Klinefelter syndrome, both of them are going to be sterile. They cannot have babies. There, for example, in Turner syndrome, she is sterile. Why? The reason being that her ovaries are rudimentary, are not been fully formed. Why? Because she just have one chromosome, one X chromosome. She ideally should be having two, but only one chromosome is there, which is exactly why her ovaries are going to be rudimentary, that means not fully formed, which is exactly why she is going to be sterile. In the case of Klinefelter syndrome, even though having a masculine body, but also having gynecomastia as in formation of breast and things like that, they will be also sterile. There is no spermatogenesis that is going to be seen in their case. Okay, and it's a genetic disorder that is there. Feminine characters, exactly. So how do you write the answer over here? Aneuploidy is a condition in which an organism has an abnormal number of chromosome, either due to gain or due to loss of more than one chromosome. It is caused by non-disjunction during cell division. What do you mean by that? Non-disjunction means no proper division of the chromosome during the cell division. So when the cell divides, obviously this many number of chromosomes should go over here, this many number of chromosomes should be going over here. That is not happening. Not disjunction of chromosomes during the cell division is not happening properly. That is why this problem is there. Example, Down syndrome, Turner syndrome. Okay, in Down syndrome, trisomy of the chromosome number 21st. In Turner syndrome, monosomy of the X chromosome is being seen. Okay, and the karyotype of an individual affected by Klinefelters is 47XXY, as we already discussed. Symptoms, underdeveloped secondary sexual characters. Yes, the secondary sexual characters are also not going to be too much developed. Gynecomastia as an enlarged breast. Infertility due to lack of spermatogenesis. Tall stature with long arms and legs. That is enough. That is be more than enough. Is that question clear to all of you? Is it clear to all of you? Clear? Not clear? Okay. So let's go ahead with the next question. Okay, and children, I hope you're doing good. Okay, my energy is a little bit less and only you can lift it up, to be honest. Okay, my energy is a little bit less. It's been so many hours since I'm standing. Actually, I was recording a one-shot video of reproductive health which I had to go today. So tomorrow it will be released for all of you guys. Make sure you watch this video. That video is definitely going to be good. Yes, so you lift up your energy. If you lift up your energy, that is going to be reflected over here. I'll also have energy. Okay, so yes. Next question. T.H. Morgan selected fruit fly for his genetical experiments. Why? Why? Give any 3 reasons. Why do you think? Hi ma'am from 2025 batch. Hi Shamant. I hope you're doing great. So there are people coming up for lifting up the energy, it seems like that. Okay, anyways, so yes, why did he collect fruit flies for his experiments? First of all, the first one is they could be grown on simple synthetic media in the laboratory. They are not like I want jam, I want burger. They were not like that. Basically, they could be grown on simple synthetic media throughout their life. Okay, next thing, they complete their life cycle in about 2 weeks. So if at all I want to see some changes, I want to see the experiments out there, see the results, I do not have to wait for years. I could see that within 2 weeks itself. Okay, 2 weeks itself. Next thing, a single mating could produce a large number of progeny flies. That means I can get more things for experiments. Okay, a single mating would be required for production of more number of offsprings. Next thing, there was a clear differentiation of sexes, that is the male and the female flies. So male and the female flies were easily distinguishable. I'll tell you a fun fact. In this particular case, the males are going to be smaller and the females are going to be larger in structure. Okay, so do note that as well. So T.H. Morgan worked on the fruit flies because of all these reasons, and fruit flies are also known as what? They are also known as the Drosophila melanogaster. They are also known as Drosophila melanogaster, and motivation is saying something because they showed sexual dimorphism. Yeah, sexual dimorphism is this part. Okay, when I say that they show clear differentiation of sexes, that is sexual dimorphism. That means morphologically they are going to be different sexes, male and female. Good one, motivation. And Shamant, I hope you are fine. Yes, okay. Now there is one more part to this question, that is, what are chromosomal disorders? Give an example. Chromosomal disorders are basically caused due to absence or excess of the chromosomes. As I spoke, it's about aneuploidy that we have to go ahead with. Okay, that we have to go ahead with. So you just have to say examples are Down syndrome and the Turner syndrome. There is not too much explanation that is there. The reason I have kept this particular question right over here is because I want to explain a few more that are going to come. From this point onwards, we're going to go to the 2-marker questions, and in the 2-marker question, one thing that have been repeated for so many times throughout is linkage recombination, linkage recombination, linkage recombination. So I wanted to explain that. Can I go ahead with the next question? Raju, Kamala is your name, is it? Okay. Dear, emasculation means removal of the male part from the flower that has both the male and the female part. Okay, guys, I hope I can go with the next question that is there. Can I? Yes, madam. Okay. So what are conclusions drawn by T.H. Morgan from the crossing experiments in Drosophila with respect to linkage? So there is a term that is coming up over here, linkage and recombination. And I'll tell you that in easy way, please understand it, very, very important thing. Okay. So suppose you have a chromosome right over here and you have another chromosome right over here, right? You have alleles, small a, small p. And then there is an allele, capital A, capital B. Cool. Now, yes. So in this particular case, and in this particular case, things are going to be different. Suppose I have small a, small b over here, capital A, capital B over here. If this chromosome is there, you know, when sexual reproduction happens in the case of human beings, there are going to be some process where the chromosomes, they come together, okay, and they cross over each other. They're going to have crossing over process, okay. They would be having a process that is known as crossing over, which leads to exchange of certain regions. Here also you will be seeing that there will be crossing over. I'm just giving you two scenarios, okay. This is scenario 1 and this is scenario 2. Now when crossing over happens over here, suppose from here the parts got exchanged, this part came over here, this part came over here. Now what will you see? When I make a new chromosome, I get something to see like this. Capital A, capital B remains like that. The gene at the top remains like that. But what happens to the lower genes out there? Small b comes over here and small a goes over there. Was this the combination that we had before? Was this the combination that we had before? Here we had capital A and small a. Now we have capital A and small b. This is what a new combination. So I can say that it has recombined. There I can see what? Recombination. And I can clearly see a lot of recombination. I can clearly see a lot of recombination right over here. Just give me a second, guys. Hello? Okay, I'm so sorry guys, there was an important call. Fine. Okay, so here I see more amount of recombination. What about this? Let me check, okay? If I cut at this part and I let it go here and here, what is going to be seen? I will see something like this. Capital B and small b moves this side. Okay, capital A and small a moves that side. The genes which are there, they are still together. The genes over here, they are still together. They have not changed as they have changed over here. So do you feel recombination is happening up there? What do you think? In scenario number 1, do you feel that recombination is there? Recombination is there. Recombination definitely is there because the top portion has changed, but the amount of recombination which is going to be seen, Baba, over here it is going to be less, over here it is going to be more. What do I conclude from this? What I am trying to make you understand from this particular thing which is there, I am trying to make you understand one very important thing, that is basically the genes which are closely located to each other, I'll say that they are showing something that is known as linkage. They are showing something that is known as linkage. And the genes which are tightly linked to each other, that is these genes that we saw here, they are tightly linked to each other, they are going to show less recombination. They will be showing less new combinations out there. On the other hand, over here when I talk about these genes, they are going to be loosely linked. Why I say that? Because they are far away from each other. They are going to be loosely linked, and in the case of loosely linked genes, do you see more recombination or less recombination? what do you see? More recombination or less recombination? They are going to show more recombination. So it's very simple to remember like this. Suppose you and your friend is there, okay, and you guys are best friends, you're very close to each other. If at all you're very close to each other, there is very less chance for a new friend to enter the group. Okay, so that means tightly linked, you guys are tightly linked, which is why there will be less recombination. On the other hand, if you and your friend are not too much close to each other, what is going to happen? There are chances someone else can also come into that particular friendship out there, right? Yes, ma'am. So there will be more recombination that will be seen. So always understand, if less linkage is there, there will be more recombination. If more linkage is there, there will be less recombination. And who gave up this particular term, that is linkage and all? It was T.H. Morgan while he did his experiment. Okay, while he was doing his experiments on the Drosophila melanogaster, he came up with this term, that is linkage, and he said that it's basically the affinity of the chromosomes or the alleles to each other that remains straight, or basically they stay together. Okay, in actual terms, I'll tell you what is the term that is being used, but I hope you have understood this part. Have you understood this part? If yes, please let me know. If no, please let me know. If there are any doubts that is there in your head and you want to ask me, please let me know. Yes, madam. Okay, so yes, now I am going to come over here and I'm going to talk about this. What are conclusions drawn by T.H. Morgan from the crossing over experiments in Drosophila with respect to linkage? So it goes like this. The higher the linkage, that means the more the linkage, the lesser is the recombination, and the lesser the linkage, the higher is the recombination. In other words, the genes which are tightly linked, which are very close, show lesser recombination, and the genes which are loosely linked, they are going to show higher amount of recombination. Linkage is inversely proportional to recombinant. Exactly. Motivation, it is going to be inversely proportional. It's not going to be the same. Perfect. Now, similar questions. See how many type of questions have come. This is one. What are the conclusions drawn by T.H. Morgan from the crossing over experiment? The next question that has come is this. In Drosophila, genes for yellow body and white eye show lesser recombination, and white eye and miniature wing show higher recombination in a cross. What is the reason for this? Okay, so they have given that the gene for, so I have to make a better one. Okay, the gene for what? Yellow body, suppose, and white eye, or I'll just change it. Yellow body gene is right over here and white eye gene is right over here. They show lesser recombination, whereas the white eye and the miniature wing show more recombination. What do you mean by that? If they are showing more recombination, that means white is over here and miniature would be on the other side. It definitely has to be on the other side, which is why if at all any exchange happens, this part goes up, this and this moves up. Suppose this comes over here, white remains there. So definitely there is going to be more variation or more recombination that is going to be there. So how do we explain this? You say that yellow body and white eye genes these genes are closely located on the same chromosome. When the genes are close, crossing over occurs rarely between them. That whenever crossing over happens, still they will not be moving away from each other. Therefore, low recombination frequency is observed. This condition is known as strong linkage. On the other hand, when you see white eye and the miniature wing genes, these genes are far apart on the chromosome. Aren't they far apart? Baba, this is here and this is over here. So crossing over occurs more frequently between the distant genes. Easily it can happen, which is why hence the higher recombination frequency is seen, and this indicates what? Weak linkage. Have you understood this? This is a very important thing. Okay, there's a question that has come that basically white and the yellow genes, they show 1.3% recombination, whereas white and miniature shows 37.3% recombination. Why? Because they are far. When I say more recombination, that means they are definitely going to be far. They are going to be having less amount of linkage. Okay, the maximum amount of recombination which is there is just going to be 50%. It is just going to be 50%. Okay, anything closer to that means they are going to be very far from each other, which is why they are going to show more amount of recombination. Got it? Is this clear? Do you have doubts? Clear? Or do we have any doubts? Got it, madam. Perfect. So yes, now yes. Differentiate between linkage and recombination. This was also once question once. Linkage, it is a tendency of genes located close on the same chromosome to be inherited together. If they are close to each other, they are going to move together only. Recombination, it is a process by which new combinations of genes are produced, okay, due to the crossing over which is there. Linkage, it reduces the chances of new gene combination, whereas recombination, it increases genetic variations. Does it increase genetic variation? Yes, it definitely does increase the genetic variations out there. So these are the points that you can clearly write and get your marks and get sorted. Okay, now there is one more question related to this, that is recombinants obtained in both Mendelian and Morgan experiment, yet the process involved are different. Comment. Listen, if at all you remember Mendel's experiment, in that there was recombination. You saw that 6 of them were non-parental. Round and green was coming, wrinkled and yellow was coming. These were non-parental character. Basically, that was also recombination, new combination. Why did that happen, and why is recombination having over here in the case of Morgan? There are two reasons. There, in the case of Mendel, it was because of independent assortment. That is why you were able to see new combination. Here, in this particular case, that is in the case of Morgan's experiment, there is sexual reproduction happening, and in the case crossing over between the chromosomes happen. And when crossing over happens, then there is exchange of chromosomes or a part of the chromosome out there, definitely you're going to find more combinations or more new combinations. Am I clear? So in Mendel's X process, recombination occurs due to independent assortment of genes on different chromosome. In Morgan's experiment, recombination occurs due to crossing over between the linked genes, okay, on the chromosome. This is a simple thing that we have discussed before. So yes, is it clear? Clear, children? Or you have doubt? Got it. I hope you have got it. Yes. Now, identify the symbols used in the pedigree analysis. So this is the pedigree thing. If at all you know about the pedigree chart, okay. So I'll tell you, this is for basically the male. This is basically the female. If at all you want to show that they are affected, you color it up. This is an affected male, okay, and this is going to be an affected it is going to be an affected female out there. Apart from it, what do you have? You have something like this. If at all it is something like this, that means the sex has not been defined. If at all it mentions like this, that means 5 people are there, 5 children are there, but their sex has not been defined over here. Phrasing, hello dear, hello. Okay, their sex is not determined. Now there is one very important thing over here that we have to notice. If it comes like this, it means that there is a normal mating between a female and a male, and suppose I give like this, that means there is a daughter that is being born. Okay, now in some cases they can give you something like this. Male is there, 2 lines and then a female. What do you mean by that? 2 lines and then a female. What do you mean by that? Any idea? Any idea which is there? When 2 lines are there, contiguous, it's known as consanguineous mating, okay? It's known as consanguineous mating, which means that mating between, relatives. It is known as consanguineous mating, and it is known as mating between relatives, okay? And can you see cases like this? Will you ever see cases like this where both of them are mating and now you have got a child who's diseased? Both of them are not diseased, but the child is diseased. Do you see that? Yes. It can happen in autosomal recessive disorders, if at all you see, in sickle cell anemia and things like that. Both the partners are carriers, but their son or daughter can have the disease out there. Can't they have the disease? Definitely they can have the disease, and it can be son, it can be daughter. Okay, so please do note about that. Similarly, you have few more questions that could be coming up. The most famous one that usually comes up is the consanguineous mating one. You should be knowing about that. Fleising also said consanguineous mating. Okay, it's consanguineous, okay? Okay, now over here I have a pedigree chart, and this pedigree chart is important. Let's go. This pedigree chart, if at all I see that here here at the top, female is affected, male is fine. Okay, female is affected, male is fine. In the next generation also I see that children are affected. In their next generation also I see children are affected. What do I mean by that? It is recessive or is it autosomal disorder? If at all you are able to see disease character in every single generation, that means it is going to be what? A dominant disorder. It is definitely going to be what? A dominant disorder. If at all you see that there is skipping of generation, for example, in this generation no one is having it, in the next generation someone is having it, again in this generation no one is having it, in the next generation someone is having it, this is going to be what? Recessive disorder. It is going to be recessive disorder. Okay? Cool? Autosomal. Oh, autosomal. Okay, I'm coming to that. Okay, just give me a second. Just give me a second. Okay. Yes. So always remember about recessive and autosomal. Now, recessive and dominant disorders are there. Every single generation dominant. If they are skipping generation, it is going to be recessive. Now going over here, talking about this particular scenario which is there. Here if at all you see the male and the female, suppose in this generation the male and the female is equally getting affected. The male and the female, it is equally getting affected, and here also I do not see much difference out there. The female is the one who is being affected. I can clearly say it is going to be what? Which disorder? It is going to be an autosomal disorder. And an autosomal disorder, the males and the females would be somewhere equally affected. It won't be showing one particular gender to be affected in more amount. So this is dominant disorder and this is autosomal. Do you have any example of autosomal dominant disorder? Because until now we have not studied about it. Autosomal recessive disorder, we studied sickle cell anemia, thalassemia, phenylketonuria. Now if you're someone who have went through your NCERT really well, probably you will be able to come up with this autosomal dominant disorder which is there. Any idea? What is that? No idea? Then probably I'll only say it is going to be the autosomal dominant disorder that we have over here is going to be myotonic dystrophy. It is known as myotonic dystrophy. Please remember it. It's the only one which is going to be dominant disorder which is there in your NCERT. There is no explanation much about it, but it is there. Then there is a disease cystic fibrosis about which they have not explained, but please do know that cystic fibrosis is an autosomal recessive disorder. Okay, they have not explained, but it's an autosomal recessive disorder. Myotonic dystrophy is a dominant one. Now there is sickle cell anemia that is here. It is autosomal recessive disorder, and here also you can see both male and female is somewhat equally affected. Okay, in the case of sex linked, usually the males are going to be affected more, and you are going to see a transmission like this. The mother will be affected from the son, then the son will be affected from the son, again the daughter is going to be affected. So you will see a crisscross pattern, female male female, or probably male female male. So that is how it usually works. But yeah, that's not, they will not complicate it to you and give it to you. Just understand that here, since they do not have in every single generation, it has to be recessive disorder, and this is the case where none of the sexes are being targeted out there. Each of them are equally affected. So this is autosomal as well. So both of them are autosomal. One is dominant, other one is recessive. I hope you had got it. Cool. I hope that works. Now we go ahead with the next thing, that is haplodiploid sex determination. By the way, it's gonna end. Okay, it's gonna end really soon, so don't worry. Haplodiploid sex determination is there. Are you aware of that? Or basically, are you aware about any sort of sex determination in the case of organisms? Can we go ahead with that, and can we just discuss it shortly? If it's okay, give me a thumbs up. If it's like done for you, Let me know. Honeybee, yes, exactly. Anyway, so we're going to talk about sex determination. There are 3 different ways where we can categorize them. Okay, one is basically male heterogamy. Another one is going to be female. And basically, the other one that we have is the haplodiploid one. Haplodiploid one. We'll go one by one. So in the male one, you have basically grasshoppers as an example. Okay. Apart from that, you have humans and Drosophila as an example. So what exactly happens over here and how they are examples for each of them? So listen to me very carefully. In the case of grasshopper, okay, in the case of grasshopper, the female one is XX and the male one is going to be XO. What do you mean by XO? There is no other chromosome over here, only one X chromosome is there. Okay, now when they fuse together, suppose this and this comes together, you get XX, female, this and this comes together, XO going to be a male, this and this comes together, X going to be a female, and this comes together, XO. But if at all we see generally, this chromosome, the female one which are there, they are only going to produce similar type of chromosome. There is no different type of chromosomes that they are going to produce. They can only contribute X. Here in this particular case, in the case of male, they can produce 2 type of chromosome. One chromosome that has X chromosome, one gamete that has X chromosome, another gamete that does not have any chromosome out there, right? So this is very, very important. Please do note that. This is going to be super important. Please definitely note that. Cool. So this is an example of male heterogamety. Why? Because males are producing 2 different type of gametes. Similarly over here we have human and Drosophila. In human and Drosophila it's very similar. Okay, males produce either X or Y chromosome. Females produce only X chromosome. So which is why again it is an example of what? Male heterogamety. Male heterogamety means if males are producing 2 type of gametes, it is known as male heterogamety. I hope that works. Now we come up with the next thing, that is female heterogamety. Can you guys guess, where do you usually see female heterogamety? In which organism do you see female heterogamety? Any guesses for female heterogamety? In which organism do you see female heterogamety? I'll give you the thing for it. So they have ZZ-ZW type of sex determination. ZZ-ZW type of sex determination, and here the females are going to produce 2 different type of gametes, whereas males are going to produce only a single type of gamete. Motivational says it's bird. Exactly. The birds which are there, they show female heterogamety. Heterogamety means different type of gametes being produced. Here they can produce either Z or they produce either W. Here they are going to produce only ZZ, the males. Now whatever you do, you get 50% of them to be females and 50% of them to be males. That is the basic logic which is there. Always remember ZW, W for females, W for women, so you can remember for females. This happens in the case of birds, and that is given according to your NCERT. Now there is haplodiploid sex determination. That is a little different. Honeybee is an example for this, as motivational said before. Okay, haplodiploid sex determination is kind of very simple. It goes like this. So over here, suppose I say the diploid number of chromosome is 32. Okay, and I say the haploid number of chromosome which is there is going to be 16. Okay, diploid is 32 and haploid is 16. When I say there is a cell which is having diploid number of chromosome, this is going to be a female. And when I say there is a cell which is having haploid, half of it, it is going to be a male. It is going to be a male. What do I mean by that and how that can be possible? Listen to me. For a female which is there, suppose it wants to form gametes. What will it undergo? Which type of division does it undergo? Is it mitosis or meiosis? If a female wants to undergo division to form gametes and it wants to make sure that the number of chromosomes over here become half. It becomes half. Which type of division will it undergo? Meiosis or mitosis? Meiosis or mitosis? Meiosis, yes. So here it undergoes meiosis, okay, and it gets cells with chromosomes which are having 16 number of chromosomes. What about male? They only have 16 number of chromosomes. They cannot undergo meiosis. Rather, they are going to undergo mitosis. They are just going to increase their cells number and they will undergo mitosis to give 16 to give 2 cells having 16 number of chromosomes. Okay. Now what happens if at all we want to make a female? Again, reductional division. Exactly. Meiosis is known as reductional division. The number of chromosomes gets reduced. Okay. Now if you want to have a female honeybee being born, what do? The male gamete and the female gamete are going to fuse together. 16 plus 16 becomes what? 32, and it is again going to be female. Ma'am, how does the male honeybee form? Listen to me very carefully. Here comes a very important term which is known as parthenogenesis. It is known as parthenogenesis, where what happens? The unfertilized female gamete, it is not going to undergo any fusion. The unfertilized female gamete over here is going to transform into what? The male. It is going to transform into a male. It is known as parthenogenesis. People get confused between parthenocarpy and parthenogenesis. Parthenocarpy happens in the case of fruit, where fruit formation happens without fertilization, so there won't be any seed. This is organism. Okay, we're talking about honeybee, where the gamete which is there, the female gamete is going to transform into what? The female gamete which is there, it is transformed into something else like this. Okay, and it is very, very important. The female gamete transforms into the male organism without undergoing fertilization. This is very, very, very important. Cool. I hope it works. Does it work? Understood? So over here, how do you write the answer? Haplodiploid sex determination mechanism is a system in which sex of an individual is determined by the number of chromosomes set it inherits in this mechanism. Okay, so males develop from unfertilized eggs and are haploid in nature. Females develop from fertilized eggs and are diploid in nature. That's all. And you have to mention all of this. If at all you do that, you will get your marks out there. It's as simple as that. Cool. Now there's a question that is, what is point mutation? Give an example for point mutation. Point mutation means what? Change in a single base pair of the DNA is known as point mutation. That means, for example, I talk about the gene which was having GAG. Here only a single base pair, this is a base pair. Suppose this changes to GUG. This is known as point mutation. And this is going to change what? Whenever a single base pair is going to be changed, definitely that is going to have some effect. A change in a single base pair of DNA is known as point mutation. A classical example for that is sickle cell anemia, where what was happening? GAG was coding for an amino acid, glutamic acid. Now it is getting converted to GUG, which is coding for what? Valine. And the whole protein structure is going to change because of that. Can you explain that? So chuttu answer. Okay, very chutku question out there. Cool. Now, finally, what is the difference between pleiotropy and polygyny? So, pleiotropy, I already told you while I was explaining about phenylketonuria. One gene, okay, one gene which is there, it is having multiple effects, as in multiple characters it is handling over here. So pleiotropy, a single gene controls multiple traits. That is known as pleiotropy. One mutation in the gene affects several characters. If one gene is affected, for example, phenylalanine hydroxylase producing gene was being affected. So what was being seen? Phenylalanine is getting accumulated. Apart from that, tyrosine is not being made. When tyrosine is not being made, there is reduced pigmentation and things like that. That is an example of pleiotropy. Polygyny means what? On the other hand, polygyny is going to be a little different. Okay, so in this case of polygyny, suppose you can imagine like this. So we have studied until now that basically one gene, okay, is responsible for one trait. If at all This particular gene is there, it is responsible for height. Only what this particular gene codes, that is responsible for height. But here things are going to change. Suppose over here I have capital A gene. Here I have capital B gene. Here I have capital C gene. Now at the end, what do you have? At the end, what do you have over here? You have AA, you have BB, you have CC. All of them are going to come together and define the nature, the phenotype, the physical appearance. That is what is it defining? It's defining, suppose, the skin color. So this combination, the people who would be having this combination of these genes will be having the darkest skin tone. The darkest skin tone. Now suppose over here, I am making it small a. It becomes capital A, small a, capital B, capital C, capital C. This is going to have lesser skin color than the previous one. What if, okay, what if, Baba, over here I am converting the whole thing? All of them will be small. What is going to happen? Still all of them, they're going to come together and they're going to show the effect. AA, small bb, small cc. All of them are showing their effect. And what effect are they showing? There will be the lightest skin tone. It is definitely going to be the lightest skin tone. Very, very, very important. Cool? Okay, here multiple genes are affecting one particular trait. There in the case of pleiotropy, one gene was affecting multiple traits. Here in polygyny means multiple genes. Multiple genes are affecting one particular character and you have to write like that. So pleiotropy, a single gene controls multiple traits. Polygyny, one trait is controlled by two or more genes. Okay, here pleiotropy, one mutation in the gene affects several characters because that gene was controlling a lot of things. Here in this case, the combined effect of many genes determine the phenotype. All the genes coming together, that's going to determine the phenotype. Example is what? Your human skin, the human height, everything is like that. That is why, okay, that is why we have different skin tones. Okay, that is why we would be having different skin tones. Example, over here we have phenylketonuria in the case of pleiotropy. That's all. Ma'am, no one, because probably they might have got better teachers somewhere else. That could be a reason, or probably they are not in a mood to prepare. Either of them. Yes. Okay. So this is how it goes. And yes, before I end up this particular live, I also want to teach one more very important thing because I think it's important for you. That matters to me, that anyone who probably is trusting me and probably watching this video feels that whatever ma'am had committed at least should be completed, right? So yes, there is one thing, that is blood group, that you need to know. Okay. The blood group, it goes like this. Listen to me. We have A blood group, we have B blood group, and we have O blood group, and we also have AB blood group. How does this happen? And what is it the example for? This is an example for multiple allelism. Okay, we use a term that is known as multiple allelism. That means multiple alleles are responsible for a character. Here you are going to find some alleles, okay? One allele is going to be IA. Another allele that you have is going to be IB. Another allele that you have is going to be IO. These three alleles are going to come, it went. These three alleles which you see over here, IA, IB, IO, are going to come in different combination to decide our blood group, which is why I say that it is an example of what? Multiple allelism. Okay, it is going to be an example of multiple allelism. How does it work? Listen, if at all I want A blood group, then the combination should be either IA. In that case, it is A blood group, or the combination could be IA and IO. If it is this combination, then you need to understand why does it show A blood group? Because A could be dominant over O. A is going to be dominant over O. So we know that A is a dominant character, right? That's an understanding. A is definitely coming under team dominant. Cool? And under team recessive, probably O could be coming up. Let's see the next thing. What about the B blood group? For B blood group, I need IB, or it can be IB, IO. Or it can be IB, IO. In this particular case, again I see that B is dominant over O. Right? B is definitely going to be dominant over O. In that particular case, over here, it is going to be what? Again, in this particular team dominant, you have got B, and here you have got O. O remains the same only, so I do not have to write it again. Okay, guys, now can you tell me, when do you see O blood group? Anyone, can you tell me, when do you usually see O blood group? Ma'am, tomorrow's schedule, motivational, tomorrow you would be having my one-shot video on reproductive health. That's for sure. I have recorded it. Now, Somik sir would be having his FA-1 physics class tomorrow at, I think, 4:30 or 5:30, something like that. Works? So, I have a question that is, what will be the genotype? Okay, what will be the genotype of a person who is having O blood group? II, exactly. II. So II, or basically you can write as IOO. But for anyone who is watching it later on, do understand II means the same thing itself. Okay, for the people who are having AB blood group, what is going to be their group? Basically it will be IA and IB, and this is very, very important. What does it show? It shows that both the alleles A and B, both of them are co-dominant. Until now we didn't study about co-dominance. This is an example of what? Co-dominance. It means that both the alleles are going to be what, Baba? Equally dominant. They are going to be equally dominant, which is why you're going to see something like this over here. Okay, very, very, very important. Fine, that is AB blood group. In AB blood group, antigen is absent. Is that what you're saying? Yes, antigen would be absent. Okay, so this is co-dominance. It's uniquely shown by A and B blood group, and yes. So my dear children, that's all. I think I have summed up everything. Is there anything that you want to ask me from this chapter, that is principles of inheritance and variation, that probably I'm missing and you want me to remind and ask me? You can ask me right now. For reproductive health, so I'm not planning to have a next marathon out there, but I have made, as I said, I have made a video already. It's a one-shot video. In that one-shot video, I have discussed all the important questions that are there, second PUC questions as well, that are there from the chapter. Make sure you check it out tomorrow. Okay, apart from it, for this particular chapter which is there, do you have any doubts that you want to ask me? You can ask me right away. I want to make sure that it's clear and done. Any doubt, they can ask you a combination of anything. Suppose a person is having A blood group and they basically cross with B blood group, what is going to be happening and things like that. Okay, codominance. Codominance is a doubt. Codominance basically means alleles. If at all you have two alleles and two alleles are equally dominant, they are going to show their characters equally, then that is an example of codominance. Just like how we had incomplete dominance, one was not able to be completely dominant, which is why it was not able to show the character. Here both of them are going to be equally dominant, that is why it is known as codominance. Mehran, okay, ma'am, take some rest. That's so sweet of you. Yes, I'm just going to take rest. Oh my God, I have been standing, I think, from 3 o'clock. Yes, from 3 o'clock. I was recording the one shot and then live. But yes, you guys kept me keep going. Okay, so I hope it's sorted. Anything else that is there, or shall we end the live? So we shall end the live, I feel, if there is no other doubts that is there. And make sure you practice some more questions out there. Check if at all you're able to answer all the questions. Okay? So that's all. Okay then, champions, thank you so much for being there in the class. Make sure you join the WhatsApp channel. Also, there's the AIO page that you can check out. Okay, in the AIO page, this particular, it's for free. You can just log in for free. The link is there in the description section of this particular video. Check that out, and basically you will be able to see this. You click over here, you will be having subject by segregation, and whatever videos are done in the YouTube, it will be put over there. It will be very easy for you to analyze and go through and watch through it. Okay, and there will be assignments sooner or later, it will be put there. Thank you so much, Mehran, and thank you so much, Motivational, and everyone out there. Lots of love to you. Please make sure you give your examination in the best possible way. And I've done one-shots for sexual reproduction, flowering plants, and also human reproduction. If you want to watch that, you can go ahead and watch it if you have confusions. Bye. Lots of love.